Hello there, a place where curiosity slows its breathing, where science takes the long way around, and where the most interesting questions are allowed to linger without demanding quick answers. Wherever you are right now, whether you're settling into bed, sitting quietly with a warm drink, or letting the day finally loosen its grip, you're invited to stay a while. Tonight's journey takes us to a place so remote, so quiet, and so carefully hidden by nature that even now, in an age of satellites and deep-sea robots, it continues to guard secrets no human eyes have ever laid upon. Before we drift too far south, it's worth pausing for a moment. Where are you listening from tonight? What part of the world holds you as these words unfold? Somewhere clocks are chiming morning. Somewhere else night has already wrapped itself tightly around the streets. And far beyond all of that, there is a continent where time itself feels suspended, where days stretch and shrink in unfamiliar ways, and where sunlight can disappear for months at a time. If you find this exploration calming, intriguing, or simply a gentle companion as you unwind, a like or a subscription helps this quiet corner reach others who might also need a slower pace. No rush, no pressure, just a small signal that curiosity still has a place to rest. Now, imagine standing at the edge of the known world, not metaphorically, but quite literally. Antarctica is not just cold, it is not merely distant. It is a continent that resists human presence at every level. The air itself pushes back, dry and sharp. The wind moves with an authority that feels almost intentional. Sound behaves strangely there, carried too far or swallowed entirely. And beneath the ice, beneath layers so thick they crush mountains flat, there are places that have never felt a footprint, never echoed with a voice, never been warmed by breath. Antarctica covers nearly 14 million square kilometres, making it the fifth largest continent on Earth. Yet it has no cities, no permanent population, no roads threading settlements together. It exists largely on its own terms. Nearly 98% of its surface is buried beneath ice, in some places more than 4 kilometres thick. This ice is not static. It flows slowly, imperceptibly, like a frozen river moving at the pace of centuries. And beneath it, hidden entirely from view, lies a world that was once exposed, shaped by rivers, oceans, forests and climates, utterly unlike what we associate with Antarctica today. Long before ice claimed the continent, Antarctica was part of a warmer Earth. Millions of years ago it supported plant life. Rivers carved valleys, shallow seas teemed with organisms. When the continent drifted south and global temperatures fell, ice advanced and sealed that ancient surface away, preserving it in darkness and pressure. What remains below is not just rock and sediment, but entire ecosystems locked off from the rest of the planet. This is where the idea of unseen creatures stops being speculation and begins to take on scientific weight. Antarctica is not lifeless. It never has been. Even in its most extreme conditions, life persists. Microbes thrive in ice, bacteria survive in subglacial lakes, and complex organisms inhabit the frigid waters along its edges. But the vast majority of Antarctica has never been sampled, not mapped in detail, not drilled into, not observed directly. Consider Lake Vostok, one of more than 400 subglacial lakes discovered beneath the Antarctic ice sheet. Lake Vostok alone lies under nearly 4 kilometres of ice and has been sealed off from the surface for at least 15 million years. When scientists first realized it existed, it raised a quiet but profound question. If water has remained liquid there for millions of years, heated by geothermal energy and insulated by ice, could life have persisted and evolved in complete isolation? When samples were eventually retrieved, they did not disappoint. DNA sequences appeared that did not match known organisms. Some were bacterial, some possibly more complex, and many remain poorly understood. This was not evidence of monsters or mythical beasts, but it was clear proof that Antarctica shelters life forms adapted to conditions humans can barely endure, let alone inhabit. And Lake Vostok is only one lake. Beneath the ice are entire networks of liquid water, connected by slow-moving currents, exchanging nutrients and heat. These environments are cold, dark and pressurized beyond anything at the surface. They have no sunlight, no photosynthesis as we know it. Life there, if complex life exists at all, would rely on chemical energy, much like organisms near deep-sea hydrothermal vents. Now shift your attention outward, beyond the buried interior, to the edges of the continent where ice meets ocean. Antarctica's ice shelves extend far over the sea, creating vast, shadowed underwater spaces where sunlight never reaches. Beneath these shelves, the ocean is quiet, stable, and cold, in a way that changes very little over time. In these regions, scientists have begun to find creatures that defy expectations: sponges taller than humans, starfish that grow slowly over centuries, fish with antifreeze proteins in their blood. These are not theoretical beings. They are documented, studied, and still deeply strange. Yet these discoveries come from only the tiniest fraction of the Antarctic marine environment. Most of it has never been seen by cameras, let alone divers. The challenge is not just cold, it is access. Ice shelves can be hundreds of meters thick. Drilling through them is expensive, risky, and limited to narrow boreholes. Sending remotely operated vehicles into the darkness beneath requires precise engineering and months of preparation. And even then, what can be observed is constrained by battery life, visibility, and sheer distance. In one remarkable expedition, scientists drilled through nearly 900 meters of ice to reach the ocean below the Ross Ice Shelf. When they lowered a camera into the black water, they did not expect to see much. Perhaps sediment, perhaps nothing at all. Instead, they found fish, amphipods, and other organisms swimming calmly in total darkness, living in an environment cut off from surface productivity. The ecosystem existed quietly, without fanfare. If such life can thrive there, it is reasonable to assume there are other organisms that have not yet been encountered. Not because they are hiding, but because we simply have not arrived. Antarctica is not unexplored because of neglect. It is unexplored because it actively resists exploration. Temperatures can drop below -60 degrees Celsius. Electronics fail. Machinery becomes brittle. Human bodies lose heat faster than they can generate it. Even reaching the interior requires aircraft capable of landing on ice, supported by temporary camps that must be dismantled before the environment claims them. Every expedition leaves behind far more questions than answers. What lives in the deepest subglacial lakes? Do multicellular organisms exist there, or only microbial life? Has isolation driven evolution in directions we've never seen elsewhere on Earth? Could there be entire branches of life that diverged long before humans walked upright, preserved by ice and time? Scientists are cautious with these questions. Speculation is measured, evidence is demanded, and yet the data we already have suggests that Antarctica is not a frozen wasteland, but a reservoir of biological possibility. A place where life has adapted to extremes not as a temporary challenge, but as a permanent condition. And then there is the time scale. Antarctica has been isolated longer than most ecosystems on Earth. Its interior has not experienced rain, open air, or sunlight in millions of years. Evolution under those conditions does not rush. It proceeds slowly, shaped by pressure, chemistry, and stability. Any creatures that exist there would not resemble familiar animals adapted to surface environments. They would not need speed, colour, or vision as we know it. They would need efficiency, endurance, the ability to survive on minimal energy for extraordinary lengths of time. This is where imagination often races ahead, but science remains steady. No one is claiming hidden giants or undiscovered predators waiting beneath the ice. The reality is quieter, but no less remarkable. Small, resilient organisms, strange forms, unusual metabolisms, life that challenges our definitions rather than our fears. And Antarctica does not exist in isolation from the rest of the scientific world. What we learn there informs how we search for life elsewhere. The icy moons of Jupiter and Saturn, like Europa and Enceladus, are believed to harbour oceans beneath thick ice shells. The conditions are not identical, but they are similar enough to make Antarctica a natural testing ground for our expectations about life in extreme environments. If life can persist beneath kilometres of ice on Earth, sustained by heat from below and chemistry rather than sunlight, then the universe may be more biologically generous than we once believed. But before we drift too far into space, it's worth staying grounded in what Antarctica already offers. A continent that humbles technology. A place where silence is not absence but presence. Where the unseen is not mythical but simply waiting. As we continue, the focus will narrow, moving closer to the specific environments where unknown life is most likely to exist, and to the reasons so much of Antarctica remains untouched even today. The ice above is thick, the distance is great, and the story beneath it has only just begun to surface. Antarctica is often described as frozen, but that word barely scratches the surface of what the continent truly is. Frozen suggests stillness, something paused, something waiting to thaw. Antarctica is not waiting. It is active in its own restrained way, moving, shifting, reshaping itself under layers of ice so vast that they quiet almost everything above them. This is a place where silence is not the absence of sound, but a physical presence, pressing down just as firmly as the cold. Standing anywhere on the Antarctic interior, the first thing that becomes apparent is scale. The horizon stretches unnaturally far, bending gently under the curve of the earth, uninterrupted by trees, buildings, or terrain features familiar to the human eye. Snow does not fall softly here most of the time. It arrives driven sideways by wind, fine grains scouring exposed surfaces until even metal yields. The ice underfoot is not uniform. It creaks, settles, and flows slowly, inch by inch, year after year, carrying the weight of history along with it. This silence is deceptive. Antarctica is not empty. It is sealed. Nearly everything that happens on this continent happens out of sight. Beneath the ice sheet, pressure increases with depth, squeezing air from snow and compressing it into ice so dense it traps ancient atmospheres inside. Tiny bubbles preserve snapshots of Earth's climate from hundreds of thousands of years ago. These frozen records tell stories of warmer periods, colder extremes, rising and falling carbon levels. They are memories held not in books, but in crystal lattices of ice. The ice itself is not a static cap. It behaves more like a slow-moving ocean. Gravity pulls it outward from the high central plateau toward the coast, where it fractures into glaciers that slide toward the sea. This movement is so gradual that it escapes casual notice, yet powerful enough to carve bedrock and transport entire landscapes beneath it. Mountains are ground down, valleys are reshaped, ancient surfaces are buried deeper, hidden more completely with each passing millennium. Above this slow-motion world, the atmosphere behaves differently too. Antarctica is the driest continent on Earth, technically a desert. Some regions receive less precipitation than the Sahara. Moisture freezes before it can fall, and snow accumulation happens so slowly that a single layer might represent decades. This lack of moisture contributes to the starkness. There is no smell of soil, no vegetation breaking the wind, no insect humming in the background. Sound travels far and strangely, bending with temperature gradients, sometimes carrying voices kilometers away, sometimes swallowing them whole. The light behaves oddly as well. During summer months the sun never fully sets. It circles the sky, low and constant, casting long shadows that rotate but never disappear. In winter, darkness takes over completely. For months there is no sunrise, no visual marker for morning or evening. Time loses some of its usual structure. Human routines falter. The body struggles to keep track of days when light offers no guidance. This environment has shaped every form of life that dares to exist here. On the surface, that life is sparse. A few hardy lichens cling to exposed rock along the coast. Moss survives where meltwater briefly appears. Tiny invertebrates endure in soil pockets during the brief summer thaw. Birds and seals visit seasonally, drawn by the productivity of the surrounding ocean rather than the land itself. To the casual observer, Antarctica seems almost sterile. But this impression changes once attention shifts downward. Below the ice sheet lies a continent that looks nothing like the white expanse above. Radar mapping and seismic surveys have revealed mountain ranges rivaling the Alps, deep rift valleys, volcanic regions, and vast basins that once held rivers and forests. The ice conceals a complex geology shaped long before polar conditions took hold. And within this buried world, conditions are not uniform. Heat rises from Earth's interior. Pressure lowers the freezing point of ice. In certain places, liquid water persists. This is how Antarctica can be both locked in ice and alive beneath it. The silence above does not mean inactivity below. Water flows slowly beneath the ice, lubricating its movement, carving channels, pooling in lakes that have no direct contact with the surface. These lakes are isolated not just from sunlight, but from the atmosphere itself. They exist under crushing pressure, in complete darkness, supplied by minerals dissolved from rock and warmed by geothermal energy. Such conditions are hostile by human standards, but life has never required comfort. It requires opportunity. In these sealed environments, any organisms that exist must operate at the edge of what biology allows. Metabolism slows. Growth takes extraordinary amounts of time. Energy is recycled with ruthless efficiency. Waste becomes resource. Nothing is wasted because nothing can be replaced easily. This is not a place for abundance or speed. It is a place for persistence, and this persistence has consequences. Isolation over geological timescales changes things. When populations are cut off for millions of years, evolution follows paths that diverge quietly but decisively. Traits that are unnecessary disappear. Traits that support survival under constant pressure remain. Over time, organisms become perfectly suited to environments that feel alien to us, but are stable to them. The Antarctic ice sheet acts as both barrier and shield. It prevents exchange with the outside world. But it also protects what lies beneath from sudden change. Surface climates shift, ice ages come and go, sea levels rise and fall. Beneath kilometres of ice, conditions remain relatively consistent. Cold, yes. Dark, absolutely, but stable. This stability is one reason scientists suspect that Antarctica may harbour life forms unlike any yet catalogued. Not because they are fantastical, but because they have had uninterrupted time to adapt without interference. Most ecosystems on Earth are shaped by constant disruption. Storms, predators, migration, competition. Antarctica's buried environments are shaped by endurance instead. The silence above reinforces this separation. Human activity barely registers here. There are no roads crossing the interior, no permanent settlements. Research stations hug the coast or sit carefully on ice, supplied by aircraft and seasonal convoys. Even these outposts are temporary in spirit. When conditions become too harsh, humans leave. Antarctica remains. And with every departure, the continent resets to quiet. This quiet matters. It means that much of Antarctica has never been contaminated by surface life. Microbial introductions are tightly controlled because even a small disturbance could alter ecosystems that have been isolated for longer than human history. Drilling projects use sterilized equipment. Water samples are extracted with extreme caution. The goal is not conquest or exploitation, but observation without interference. Despite these precautions, the challenges are immense. Instruments freeze, batteries fail, communication drops out. Even data retrieval can take years. A sample drilled from beneath the ice might represent the only physical contact humans ever have with that environment. One chance to learn something meaningful without damaging what has survived undisturbed for ages. This limitation has slowed discovery, but it has also sharpened focus. Every result is studied carefully. Every anomaly is examined rather than dismissed. When unusual chemical signatures appear, they are not brushed aside as errors. When DNA sequences do not match known organisms, they are preserved, reanalyzed, debated. Silence amplifies curiosity. There is also a psychological effect to this environment. Scientists who work in Antarctica often describe a heightened awareness of their own presence. Every movement feels intrusive against the stillness. Every sound carries. The awareness that beneath their feet lie worlds untouched by human eyes creates a sense of restraint rather than urgency. This mindset shapes exploration. Antarctica is not a frontier in the traditional sense. It is not something to be claimed or mastered. It is something to be approached carefully, with patience and humility. The ice does not yield easily, and it does not forget disturbance quickly. And yet, for all this caution, progress continues. New radar techniques reveal finer details beneath the ice. Autonomous probes venture where humans cannot. Data accumulates slowly, building a picture of a continent far more complex than its surface suggests. What emerges from this picture is not a single hidden ecosystem, but many. Some connected, some isolated, some ancient, some perhaps more recently formed, all shaped by the same overarching reality. Ice above, rock below, water trapped in between. This locked world does not announce itself. It does not produce obvious signs of life at the surface. There are no forests breaking through the ice, no plumes of steam advertising warmth below. Everything is subtle, everything is indirect, and that is precisely why so much remains unseen. As technology improves and methods become more refined, scientists are beginning to piece together how these buried systems function and how life might fit within them. But understanding comes slowly here. Antarctica does not reward haste. It rewards attention. The silence is not emptiness. It is compression, a layering of time, pressure, and possibility held in place by ice that has no intention of melting anytime soon. And as the focus shifts further beneath that ice, toward the specific environments where life has already proven its resilience, the question becomes less about whether unseen creatures exist and more about how many forms of life we have yet to recognize simply because they do not resemble what we expect. The world locked in ice is not asleep. It is simply very, very quiet. When people imagine Antarctica, they tend to picture a surface that defines the whole place. Endless, white, wind-scoured plains, ice cliffs breaking into the sea. It feels complete in its emptiness, as though there is nothing more to say once you've described what lies on top. But the ice is not the story. It is the cover. What lies beneath it is older, heavier, and far more complex than the frozen shell above ever suggests. The Antarctic ice sheet is so thick, so persistent, that it erases almost every visual hint of what came before it. In some regions, the ice has remained intact for more than 30 million years. Snow fell, compressed, froze, and was buried under more snow until the original landscape disappeared entirely from view. Valleys were filled, peaks were smoothed, rivers vanished without a trace. The ice did not destroy these features, it hid them. Modern science has only recently begun to see through that concealment. Using radar that penetrates ice, seismic waves that bounce off buried rock, and gravity measurements that reveal mass below the surface, researchers have slowly reconstructed the shape of the continent underneath. What they have found is not a simple flattened land, but a terrain as dramatic as any on Earth. There are mountain ranges buried beneath kilometres of ice, some taller than those in Europe. There are deep basins where ancient seas once pooled. There are rift systems where the crust of the planet stretched and thinned, allowing heat from below to rise closer to the surface. Antarctica, it turns out, is geologically alive, even if that activity is muffled by ice. One of the most striking discoveries is the Gambutsev subglacial mountains. Completely hidden beneath the East Antarctic ice sheet, these mountains rival the Alps in height. They were unknown until the late 20th century. No peaks break the surface, no shadows hint at their presence. Yet beneath the ice they rise sharply, shaping the flow of glaciers above them and influencing where ice thickens or thins. These mountains did not form recently. They are ancient, likely hundreds of millions of years old, remnants of a time when Antarctica was part of a much warmer supercontinent. Their continued existence beneath the ice speaks to how effectively the ice preserves what lies below. Erosion still happens, but slowly. Change unfolds on timescales that make human history feel like a brief interruption. Elsewhere beneath the ice are broad, flat plains that once supported rivers flowing toward ancient coastlines. Sediment layers suggest long periods of stability, followed by dramatic shifts as climates cooled and ice advanced. Each layer tells part of a story about Earth's past, but they also hint at something else. Where there was water once, there may still be water now. Pressure plays a critical role here. Ice exerts immense force on the rock beneath it. As pressure increases, the melting point of ice decreases. Add geothermal heat rising from Earth's interior, and pockets of liquid water can persist even in temperatures far below freezing. This is why Antarctica contains hundreds of subglacial lakes, some small, some enormous, scattered beneath the ice sheet like hidden mirrors. These lakes are not all the same. Some are shallow and transient, filling and draining slowly over time as water migrates beneath the ice. Others are deep and stable, sealed off for millions of years. They're connected by networks of channels that allow water to flow from one region to another, lubricating the base of the ice sheet and influencing how glaciers move toward the sea. This movement is slow, but it is relentless. Ice above slides over wet rock below, reshaping the hidden landscape and redistributing heat and nutrients. From a biological perspective, this matters. Moving water can carry dissolved minerals, chemical energy, and potentially living organisms between isolated environments. Even in darkness, even under pressure, there can be exchange. Where there is exchange, there is opportunity. The absence of sunlight below the ice means that any life present cannot rely on photosynthesis. Instead, it must draw energy from chemical reactions, a process known as chemosynthesis. This kind of metabolism exists elsewhere on Earth, most famously near deep-sea hydrothermal vents, where bacteria use sulfur, methane or hydrogen as energy sources. Antarctica's subglacial environments share some similarities with these deep-sea systems, though they are not identical. The chemistry is different. The temperatures are lower. The energy input is smaller and more stable. Life here, if complex, would need to be exceptionally efficient, adapted to scarcity and consistency rather than abundance and fluctuation. What has already been found supports this idea. Microbial communities recovered from subglacial water samples show signs of active metabolism. They are not dormant relics frozen in time. They are living, processing nutrients, reproducing slowly. Some appear to feed on iron compounds released from bedrock. Others may use hydrogen generated by water-rock interactions deep below. These organisms are small, but they matter. They prove that Antarctica's buried environments are not sterile. They demonstrate that life can persist in isolation, drawing energy from the planet itself rather than the sun. And they raise a more intriguing possibility. If microbial life can survive and evolve under these conditions, what else might exist alongside it? Complex life requires more energy, more structure, and often more time. But Antarctica has time in abundance, millions of years without surface disruption, millions of years of relative stability. If there were pathways for simple organisms to gradually become more complex, this is the kind of environment where such slow evolution could occur unnoticed. Scientists are careful here. There is no evidence yet of multicellular organisms living beneath kilometers of Antarctic ice. But absence of evidence is not evidence of absence, especially when sampling has barely scratched the surface. Drilling operations reach narrow points in a vast buried world. What lies beyond those points remains unseen, and then there is the ocean beneath the ice shelves. Where the ice sheet meets the sea, it often extends outward, floating on the ocean surface while still attached to the land. These ice shelves can be hundreds of meters thick and stretch for hundreds of kilometers. Beneath them lies a hidden marine environment unlike any other on Earth. No sunlight penetrates. Water temperatures hover near the freezing point. Currents move slowly, bringing nutrients from the open ocean and from beneath the ice. For a long time, scientists assumed these environments would be nearly lifeless. Without sunlight, without direct contact with surface productivity, it seemed unlikely that much could survive. That assumption has proven wrong. When cameras were first lowered beneath Antarctic ice shelves, they revealed communities of organisms anchored to the sea floor. Sponges, anemones, and other filter feeders clung to rocks, growing slowly, feeding on organic material drifting in from distant waters. Some of these organisms may live for centuries. Growth is measured in millimeters per year. Change is almost imperceptible. These discoveries came from limited access points. Most ice shelves have never been explored below. The ocean floor beneath them remains largely unmapped, and where conditions differ slightly, where geothermal heat warms the water, or where nutrient flows are stronger, ecosystems could look very different from what has been observed so far. This brings us back to the idea of creatures no human has ever seen. Not dramatic beasts lurking in shadows, but organisms adapted so completely to their environment that they escape our expectations. Forms shaped by darkness, pressure, and patience rather than speed or aggression. Life that persists by enduring rather than expanding. The difficulty in finding such life is not just technological, it is conceptual. Human exploration has always been guided by what we think life should look like. We search for movement, colour, activity. In Antarctica's buried worlds, life may be subtle. It may blend into its surroundings. It may operate on timescales that make observation challenging. A creature that moves once a day would be easy to miss. One that grows over centuries might appear unchanged from one expedition to the next. One that feeds on chemical gradients invisible to our senses might leave no obvious trace. And yet, these environments are not static. Water moves, heat fluctuates, chemistry shifts. Change occurs slowly, but it occurs. Evolution responds, not with dramatic leaps, but with incremental adjustments that accumulate over vast spans of time. Antarctica's ice acts as a filter, allowing only certain influences to pass through. It blocks sunlight, it dampens temperature swings, it isolates ecosystems from surface disruptions. What remains below is a refined version of Earth's processes, stripped down to essentials. This refinement is what makes the continent so valuable scientifically. Studying life beneath the ice helps researchers understand the limits of biology. It tests assumptions about what environments are habitable. It informs how we search for life on other worlds, particularly icy moons where similar conditions may exist. But Antarctica is not a laboratory designed for convenience. Every attempt to explore beneath the ice carries risk. Drilling must avoid contaminating pristine environments. Equipment must withstand pressure, cold, and isolation. Logistics alone can take years to arrange. As a result, exploration proceeds cautiously, deliberately. Each mission builds on the last, expanding our understanding incrementally. Each discovery opens new questions rather than closing them. What is the full extent of the subglacial water network? How isolated are individual lakes and channels? Do organisms migrate between them, or are populations completely cut off? Has evolution taken unique paths in different regions, producing distinct biological communities adapted to subtle variations in chemistry and heat? And perhaps most intriguingly, are there thresholds beyond which complexity emerges? Places where energy availability crosses a line that allows multicellular life to persist, even if slowly, even if sparsely? These questions remain unanswered not because they are unimportant, but because Antarctica does not give up its secrets easily. The ice above never truly melts. It thickens in some places, thins in others, but it remains. A lid on a world that has learned to exist without being seen. As exploration continues, attention increasingly turns to how water, rock, and life interact beneath the ice, and to the possibility that Antarctica's most profound biological discoveries are not at its edges, but deep within its hidden interior. Far from the wind-scoured surface, far below the white plains that seem to stretch forever, Antarctica meets the sea in a way few people ever picture. The continent does not end neatly at the coastline. Instead, it reaches outward, its ice sliding slowly off the land and floating over the ocean, forming vast ice shelves that act like frozen roofs over an entirely separate world. Beneath these shelves lies a dark ocean, sealed from sunlight, muffled by hundreds of meters of ice, and shaped by forces that operate quietly and relentlessly. This hidden ocean is not a small or marginal feature. Ice shelves fringe nearly 3/4 of Antarctica's coastline. Some of them are larger than the entire countries. The Ross Ice Shelf alone covers an area roughly the size of France. Beneath it, seawater flows in slow, looping currents, interacting with the underside of the ice and the seafloor below. For much of human history, this space might as well not have existed. There was no way to see it, no practical way to reach it, and little reason to suspect it held anything of interest. That assumption has changed. To understand why this ocean matters, it helps to consider how unusual it is. In most marine environments, sunlight is the foundation of life. Phytoplankton near the surface convert light into energy, forming the base of food webs that support fish, mammals, and everything else. Even in the deep sea, far below the reach of light, ecosystems are usually sustained by organic matter drifting down from above. The dark ocean beneath Antarctic ice shelves receives neither direct sunlight nor consistent surface input. It exists under a lid that blocks photosynthesis entirely, and yet it is not empty. The water beneath the ice shelves is cold, hovering just below the freezing point of seawater, but it remains liquid due to pressure and salinity. It is also surprisingly stable. Temperatures change slowly, currents move at measured speeds. Storms above have little effect below. In many ways, this environment is calmer than the open ocean, even if it is far more difficult to access. Where water flows, life often follows. The key question has always been how energy enters this system. Some of it arrives indirectly. Ocean currents can carry organic particles from distant sunlit regions, filter-feeding organisms anchored to the seafloor. Some energy comes from chemical reactions between seawater and sediments. In a few locations, geothermal heat seeps upward, slightly warming the water and altering its chemistry. These inputs are small compared to surface oceans, but they are steady. Over long periods, steadiness can matter more than abundance. The first glimpses into this hidden ocean came through narrow drill holes melted through the ice. Scientists lowered cameras and instruments into the darkness, unsure of what they would find. In some cases, they expected nothing more than sediment and perhaps a few microbial mats. Instead, the cameras revealed living communities, sponges attached to rocks, starfish moving slowly across the sea floor, worm-like creatures burrowed into soft sediments. These were not isolated individuals carried in by chance. They formed structured ecosystems with organisms occupying specific niches, interacting in ways familiar to marine biologists, despite the extreme conditions. What was most striking was not just their presence, but their apparent age. Growth rates in these cold, dark waters are incredibly slow. Some Antarctic sponges are thought to live for hundreds or even thousands of years. Their tissues accumulate chemical signatures that record environmental conditions over centuries. They are living archives, shaped by patience rather than competition. These organisms did not evolve recently. Many likely trace their ancestry back to a time when Antarctica was warmer, when ice shelves were smaller or absent, and when sunlight reached areas that are now permanently dark. As the ice advanced, Some lineages adapted rather than retreating. They adjusted their metabolisms, slowed their growth, and survived as the world above them froze. But adaptation is rarely static. Isolation drives change. Over millions of years, populations beneath ice shelves have been separated from their relatives in open oceans. Gene flow has been restricted. Selection pressures have shifted. In such circumstances, divergence is inevitable. This raises an intriguing possibility. Some organisms beneath the ice shelves may represent distinct species, or even entire lineages, that exist nowhere else on Earth. Not because they are dramatic or exotic, but because they have quietly followed their own evolutionary paths unnoticed. The difficulty is that we have barely sampled these environments. Most observations come from a handful of drill sites. Each site represents a tiny window into a vast underwater landscape. Between these points lie hundreds of kilometers of seafloor that have never been seen, let alone studied. And then there is the underside of the ice itself. The base of an ice shelf is not smooth. It is sculpted by melting and refreezing, forming channels, ridges and cavities. These structures influence how water moves and how nutrients are distributed. In some places, melting releases fresh water into the ocean below, creating layers of different salinity that affect circulation. In others, freezing extracts fresh water, increasing salinity and causing water to sink. These processes create microenvironments, subtle variations in temperature, chemistry, and flow that can shape where organisms live. A slight increase in nutrient availability or a small change in current speed can make the difference between survival and absence in such energy-limited systems. Life responds to these gradients even if we struggle to detect them. There is also the question of mobility. Many organisms beneath ice shelves are sessile, anchored to the sea floor, but others are mobile, albeit slowly. Fish have been observed swimming beneath the ice, navigating in complete darkness. Their eyes are often small or adapted to low light. Some rely more on lateral line systems to sense movement and pressure changes in the water. These fish raise questions about how animals orient themselves in such environments. Without visual landmarks, without daily light cycles, navigation must rely on other cues. Chemical gradients, water currents, perhaps even subtle vibrations transmitted through the ice above could play a role. Predation exists here too, though at a subdued pace. Energy scarcity favours efficiency. Chases are rare. Ambush and scavenging are more common. Life unfolds slowly, measured not in bursts of activity, but in long intervals of waiting. The deeper one looks, the more this ocean begins to resemble an alien world, not because it defies biology, but because it operates under a different set of priorities. Speed is unnecessary. Reproduction may be infrequent. Longevity becomes a strategy rather than an accident. This is where the idea of unseen creatures becomes most compelling. In such environments, organisms do not need to be large or aggressive to be successful. They need to endure. They need to use what little energy is available with precision. Over time, this can produce forms that look unfamiliar, not monstrous, but subtly different in structure and function. Soft-bodied organisms that leave little fossil record, transparent or pale species that do not invest in pigmentation, slow-growing animals whose changes are difficult to notice across human lifetimes. These are the kinds of creatures most likely to remain undocumented. Exploration of the dark ocean beneath ice shelves is constrained by technology. Drilling through ice is expensive and time-consuming. Remotely operated vehicles must be lowered through narrow shafts, limiting their range of movement. Communication is delayed or intermittent. Each mission must prioritize specific questions, leaving countless others unanswered. Even when organisms are observed, collecting samples can be risky. Removing individuals from such stable environments risks disrupting fragile systems. Ethical considerations weigh heavily on decisions about what to take and what to leave undisturbed. As a result, much of what scientists know comes from images, chemical measurements, and indirect evidence. DNA fragments recovered from water samples hint at biodiversity that has not been visually confirmed. Chemical signatures suggest metabolic processes that imply the presence of organisms yet to be seen. There are moments when instruments detect movement or signals that cannot be easily explained. These are not dramatic anomalies, but subtle inconsistencies, slight changes in particle concentrations, unexpected shifts in chemical gradients. They prompt further investigation, not speculation, but they remind researchers that the picture is incomplete. This incompleteness is not a failure. It is a reflection of the scale and inaccessibility of the environment. The dark ocean under the frozen shell is not designed to be known quickly. It resists mapping and cataloguing. It reveals itself in fragments. What makes this ocean particularly important is how it connects to broader questions about life on Earth and beyond. Similar ice-covered oceans are thought to exist on moons like Europa and Enceladus. Studying how life persists beneath Antarctic ice shelves informs how we might search for life in those distant places. If ecosystems can survive here, drawing energy from chemistry and distant nutrient sources, then the requirements for life may be broader than once assumed. Antarctica becomes not just a place of discovery, but a reference point. At the same time, this knowledge carries responsibility. As climate change alters Antarctica's ice shelves, thinning them, and in some cases causing them to collapse, the dark ocean beneath is exposed to new conditions. Sunlight reaches areas that have been dark for millions of years. Currents change, temperatures shift. These changes may allow new species to move in, altering ecosystems that evolved in isolation. They may also threaten organisms adapted to stable cold conditions. Understanding what lives beneath the ice now is essential to understanding what might be lost. For now, much of this ocean remains unseen. Cameras have skimmed its surface. Samples have been taken from a few locations. Vast stretches lie untouched. Their inhabitants living out slow lives in darkness, unaware of the attention their world has begun to attract. As exploration continues, focus will turn toward how these hidden marine systems connect with the subglacial environments further inland, and how water, life, and ice form a continuous network beneath the frozen shell. The boundary between land and sea blurs here, replaced by a layered system where ice, water, and biology intertwine in ways we're only beginning to trace. Life on Earth is so often explained through a single, comforting framework that it can be difficult to imagine alternatives. The sun rises, plants capture its energy, animals eat the plants or each other, and everything else follows from that steady input of light. Seasons turn, days lengthen and shorten, biological clocks tick in time with the sky. This pattern feels so fundamental that it becomes invisible, like gravity or air. Yet beneath Antarctica's ice, and beneath its floating shelves, this familiar rhythm simply does not exist. Here, sunlight never arrives. There are no dawns, no dusks, no summers marked by abundance or winters marked by scarcity. There is only darkness, cold, pressure, and time stretched into something far less dramatic than change. And still, life persists. To understand how life functions without sunlight or seasons, It helps to step away from surface intuition. Photosynthesis is not a requirement for life itself. It is only one strategy among many, albeit a highly successful one. Long before plants colonized land, and long before oxygen filled the atmosphere, life on Earth survived using chemistry alone. In some of the planet's earliest environments, energy came from reactions between water and rock, from volcanic gases, from gradients in temperature and composition. Those pathways never disappeared. They were simply overshadowed by the efficiency of sunlight. Antarctica's hidden environments bring those older strategies back into focus. Without sunlight, there is no photosynthesis, which means no plants, no algae, no phytoplankton forming the base of a classic food web. Instead, the foundation of life here is chemosynthesis. Certain microorganisms are able to extract energy from chemical reactions involving inorganic compounds. Iron, sulfur, hydrogen, methane, and nitrogen compounds become fuel rather than waste. These microbes convert chemical energy into biological matter, forming the base of ecosystems that operate entirely out of sight. This process is slow. Chemical energy yields less usable power than sunlight. As a result, life in these environments runs at a different pace. Growth is measured in years rather than days. Reproduction may occur infrequently. Metabolic rates are reduced to conserve energy. In many cases, organisms enter long periods of near dormancy, remaining alive but minimally active until conditions favour even slight growth. In the absence of seasons, there are no predictable cycles of abundance and decline. At the surface, seasonal change drives behaviour. Animals migrate, plants flower, insects emerge. Beneath the Antarctic ice, none of this applies. Conditions remain relatively constant year after year, century after century. Temperature fluctuates only slightly. Nutrient input is steady but limited. There is no signal telling life when to speed up or slow down. This constancy reshapes biology in profound ways. Organisms that live here do not need internal clocks tuned to light cycles. Circadian rhythms, so important for most surface life, become irrelevant. Instead, biological timing may be regulated by internal chemical states or by extremely subtle environmental cues, such as changes in water chemistry or pressure. Some organisms may not follow cycles at all, operating continuously at low intensity. Reproduction under these conditions is particularly challenging. Producing offspring requires energy, and energy is scarce. Many organisms likely reproduce slowly, investing heavily in each reproductive event. Eggs may develop over long periods. Larval stages may be abbreviated or eliminated entirely. In some cases, reproduction may occur only when conditions align just right, perhaps triggered by a slight increase in available nutrients or a rare disturbance that redistributes resources. Longevity becomes a survival strategy. Instead of growing quickly and reproducing often, organisms benefit from living a long time, maintaining themselves efficiently and reproducing sparingly. This leads to populations composed of individuals that may be decades or centuries old. Such timescales are difficult for humans to grasp, but they make sense in an environment where change is rare and risk is high. Without sunlight, visual systems lose their advantage. Eyes become less useful, and maintaining them costs energy. Some organisms retain simple light-sensitive structures, capable of detecting faint glows or bioluminescence, but many rely on other senses. Chemical detection becomes crucial. Being able to sense gradients in dissolved compounds can help organisms locate food, mates, or suitable habitats. Mechanical sensing also plays a role. Pressure waves, Vibrations and water movement carry information in darkness. Fish and invertebrates may rely on lateral line systems or similar sensory networks to perceive their surroundings. These systems detect changes in water flow and pressure, allowing navigation and interaction without vision. Communication, if it occurs at all, is subtle. There are no bright displays or rapid movements designed to attract attention. Signals are likely chemical or mechanical, tuned to short distances and low energy expenditure. Even predation follows these constraints. High-speed chases are inefficient. Ambush, scavenging, and opportunistic feeding dominate. In such an environment, competition takes on a different character. On the surface, competition often favors speed, aggression, and rapid reproduction. Beneath the ice, competition favors efficiency, resilience, and the ability to endure long periods with minimal input. Species that waste energy do not persist. Those that conserve it do. One of the most striking implications of life without seasons is evolutionary stability. On the surface, environmental variability drives rapid adaptation. Ice ages, droughts, floods, and temperature swings constantly reshape ecosystems. Beneath Antarctica's ice, conditions have remained remarkably stable for millions of years. This stability allows evolutionary change to proceed slowly, guided by subtle pressures rather than dramatic shifts. Over such long periods, even small selective advantages can accumulate. Traits that improve energy efficiency by a few percent become decisive. Slight changes in enzyme function, membrane composition, or metabolic pathways can determine survival. These changes may not produce dramatic differences in appearance, but they can fundamentally alter how organisms function. This is one reason scientists suspect that Antarctica's hidden environments may host life forms that are biologically unfamiliar, even if they do not look particularly strange. Their novelty may lie in their chemistry rather than their shape. Proteins adapted to function under constant cold and high pressure, cell membranes that remain flexible without heat, metabolic pathways optimized for extremely low energy flux. Such adaptations have already been observed in Antarctic organisms close to the surface. Fish in icy waters produce antifreeze proteins that prevent their blood from crystallizing. Enzymes in cold-adapted microbes function efficiently at temperatures that would halt most biochemical reactions. Beneath the ice, these adaptations may be taken even further. The absence of seasons also affects population dynamics. On the surface, populations often fluctuate dramatically, rising and falling with food availability and climate. Beneath the ice, populations are likely small and stable. There is little opportunity for rapid expansion. Disturbances are rare. When change does occur, recovery may take centuries. This fragility makes these ecosystems particularly sensitive to disruption. Introducing new organisms, altering nutrient flows, or changing temperature even slightly could have outsized effects. Species adapted to constant conditions may struggle to cope with variability. This is one reason why scientists are cautious about exploration and emphasize contamination control so strongly. There is also the question of how life here originated. Did organisms colonize these environments before the ice advanced, gradually adapting as conditions changed? Or did life enter later, migrating downward through cracks, channels, or water networks beneath the ice? The answer may differ from one location to another. In some cases, geological evidence suggests that subglacial lakes and marine environments were once connected to the open ocean. As ice shelves formed and thickened, these connections narrowed or closed, trapping organisms inside. In other cases, water bodies may have formed entirely beneath the ice, creating new habitats that life colonized from below via groundwater or from above via ice-penetrating channels. Once isolated, these populations would have followed their own evolutionary trajectories. Genetic drift, local adaptation, and the absence of competition from surface species would shape their future. Over millions of years, divergence would become inevitable. The lack of seasons also alters how organisms respond to time. Without external cycles, internal processes may become decoupled from the calendar entirely. Aging may proceed differently. Repair mechanisms may be emphasized over growth. Damage accumulates slowly, but repair systems must be efficient to maintain function over long lifespans. Some researchers speculate that organisms in these environments may have unusually robust DNA repair mechanisms, protecting their genetic material over centuries. Others suggest that low metabolic rates reduce the accumulation of damage in the first place. Either way, these strategies offer insights into fundamental biological questions about longevity and resilience. Life without sunlight also forces a re-evaluation of what constitutes productivity. Surface ecosystems are often judged by how much biomass they produce. Antarctic sub-ice ecosystems produce very little biomass, but what they produce is persistent. Matter is recycled repeatedly. Waste becomes food. Nothing moves quickly through the system, but little is lost. This recycling creates tightly coupled communities where changes ripple slowly but thoroughly. Removing a single species could have cascading effects that take generations to manifest. Conversely, adding a new energy source could restructure the entire system over time. From a human perspective, these ecosystems feel almost invisible. They do not announce themselves. They do not align with our expectations of life. But they are not marginal. They represent a mode of existence that has always been part of Earth's biological repertoire, simply overshadowed by the success of sunlit life. Antarctica brings that mode back into view. As research continues, scientists are beginning to piece together how these dark, seasonless ecosystems function as integrated wholes. Water flow connects inland lakes to coastal oceans. Chemical gradients link rock to biology. Ice shapes everything from habitat structure to nutrient availability. This interconnectedness suggests that life beneath Antarctica's ice is not confined to isolated pockets, but forms a continuum extending from the continent's interior to the dark ocean beneath its shelves. Understanding this continuum is essential to understanding how unseen creatures might exist, persist, and interact. And as attention shifts toward environments even deeper beneath the ice, where pressure increases and energy becomes even scarcer, the question is no longer whether life can exist without sunlight or seasons. That question has been answered. The question now is how far those limits extend and what forms life takes when stripped of nearly every familiar reference point, guided only by chemistry, patience, and time. There is a particular phrase that appears again and again in scientific papers about Antarctica, usually tucked into introductions or discussion sections, almost as a quiet aside. It goes something like this: conditions previously thought to be uninhabitable. The wording is careful, understated, and repeated often enough that it loses its drama. But when you pause and unpack what it actually means, it becomes clear that Antarctica has spent decades gently, persistently correcting our assumptions about where life can exist. For a long time, the boundaries of habitability were drawn with confidence. Life needs warmth. Life needs light. Life needs access to nutrients in predictable cycles. Remove enough of those factors and biology simply stops. Antarctica has been methodically dismantling that logic, not through spectacular revelations, but through steady accumulation of evidence found in places where nothing was expected to survive. The first surprises came from the most obvious extreme: cold itself. In the exposed regions of Antarctica, particularly the dry valleys, temperatures remain below freezing for most of the year, and liquid water is scarce to the point of near absence. These valleys are scoured by wind, deprived of snow accumulation, and blasted by ultraviolet radiation reflected from ice and clouds. By almost any intuitive measure, they should be lifeless. They are not. Microbial communities exist within rocks, not on them, but inside them. Tiny cracks and pores in translucent stones allow a small amount of light to penetrate, just enough for photosynthetic microbes to function. These organisms are called endoliths, and they live protected from the worst of the cold and radiation by the rock itself. Their metabolism is extraordinarily slow. They may divide only once every several years. Some may go dormant for decades at a time. What matters is not speed, but persistence. As scientists pushed further into Antarctic extremes, they began to find life not just sheltered from harsh conditions, but operating fully within them. Beneath glaciers, microbial communities thrive in meltwater channels at the ice-bed interface. These organisms do not rely on sunlight at all. They feed on iron, sulfur, and other elements released as ice grinds rock into fine powder. The grinding itself provides fresh surfaces for chemical reactions, creating energy sources that microbes can exploit. In these dark environments, bacteria form the base of food webs that extend upward in unexpected ways. Tiny invertebrates, such as nematodes and tardigrades, have been found in Antarctic soils and ice-associated habitats. These creatures are famous for their resilience, capable of surviving freezing, dehydration, and radiation that would kill most animals. In Antarctica, they do not merely endure, they live, feed, and reproduce. Some nematode species found there have life cycles measured in years rather than weeks. They enter cryptobiotic states when conditions become too harsh, effectively suspending their metabolism. When water becomes available again, even briefly, they resume activity as if nothing happened. This ability blurs the line between living and waiting, a theme that appears repeatedly in Antarctic biology. Moving deeper into subglacial environments, the discoveries become even more striking. Water samples drawn from beneath kilometers of ice contain diverse microbial communities. These are not contaminants introduced during drilling. Careful controls and repeated sampling have confirmed their origin. The organisms show genetic adaptations to cold, pressure, and low energy availability. Some are related to known bacteria, but others diverge significantly, hinting at long periods of independent evolution. In Lake Whillans, a subglacial lake beneath the West Antarctic ice sheet, scientists found microbes that actively metabolize nitrogen, sulfur, and iron. These chemical cycles resemble those found in deep-sea vents and subsurface aquifers elsewhere on Earth, but here they operate beneath ice that has sealed the environment for hundreds of thousands of years. The organisms are not relics frozen in place. They are part of a functioning ecosystem. What is particularly notable is that these ecosystems do not appear to be barely hanging on. Chemical measurements suggest steady activity. Metabolic byproducts indicate ongoing processes rather than sporadic bursts. Life here is slow, but it is not fragile. Then there are the creatures found beneath the floating ice shelves, in the dark ocean already touched on earlier. In one expedition beneath the Filchner-Ronne ice shelf, researchers discovered a sponge-like organism attached to a rock more than 1,000 kilometers from open water. There was no obvious source of food nearby, no sunlight, No clear explanation for how such an organism could sustain itself in that location. This find prompted intense discussion. Sponges are not microbes. They are multicellular animals with complex structures and specific nutritional needs. Their presence suggested that organic matter was reaching this site through pathways not yet fully understood, perhaps via slow-moving currents carrying particles from distant regions, or through local chemosynthetic production. The implication was unsettling in the best possible way. If a sponge could live there, what else might be living out of sight? Antarctic fish provide another example of life thriving where it seemingly should not. In the frigid waters surrounding the continent, ice fish lack hemoglobin, the molecule that carries oxygen in the blood of most vertebrates. This seems like a disadvantage, yet in oxygen-rich cold water it works. Their blood is clear, and their circulatory systems are adapted to move large volumes of it efficiently. They also produce antifreeze proteins that prevent ice crystals from forming in their tissues. These adaptations are not recent. Genetic evidence suggests they evolved over millions of years as Antarctic waters cooled. The fish did not flee as conditions worsened. They adapted in place. In doing so, they explored physiological territory few other vertebrates occupy. Some Antarctic invertebrates show similar patterns. Starfish and sea urchins grow slowly, live long lives, and tolerate temperatures that would be lethal to related species elsewhere. Their reproductive strategies are adjusted to the cold. Larval development can take years. Some species brood their young, avoiding the risks of releasing larvae into a harsh environment. Each of these examples adds to a growing realization. Life does not require comfort. It requires consistency, opportunity, and time. Antarctica provides all three, albeit in extreme forms. Perhaps the most counterintuitive discoveries come from within the ice itself. Glacial ice is not inert. It contains microscopic channels where liquid water persists even at sub-zero temperatures. Within these channels, microbes survive, feeding on trapped organic material and minerals. Some ice cores contain viable bacteria that may be hundreds of thousands of years old. When warmed and provided with nutrients, they resume activity. This raises profound questions about dormancy and survival. If organisms can persist for such long periods in ice, what other environments might harbour life in similarly suspended states? Antarctica becomes a proving ground for ideas that extend far beyond Earth. But the focus here remains grounded. These are not hypothetical creatures. They are real, documented organisms found in places that once carried labels like sterile, dead, or biologically irrelevant. Each discovery has forced a revision of those labels. Importantly, many of these creatures are not rare exceptions. They are part of stable communities. Where one organism exists, others often follow, linked by shared chemistry and interdependence. Bacteria support invertebrates. Invertebrates process material that feeds other microbes. Nutrients cycle slowly but reliably. This cycling is often invisible without careful measurement. There are no blooms, no sudden population explosions. Everything unfolds at a pace that resists dramatic observation. It is easy to overlook such systems if one expects life to announce itself. That expectation has been one of the greatest obstacles to understanding Antarctica's biology. Humans tend to associate life with movement, colour, sound. We look for things that resemble ourselves or the ecosystems we know. Antarctic life challenges that bias. It is quiet. It blends into its environment. It persists rather than competes. The creatures found where nothing should live often share this trait. They do not push boundaries aggressively. They settle into them patiently. As more extreme environments are explored, this pattern repeats. Beneath sea ice, within brine channels so salty they would desiccate most cells, microbes flourish. In sub-zero soils with almost no organic matter, microscopic animals feed on bacteria and fungi. In water trapped under pressure for geological timescales, metabolic networks persist. Each of these discoveries carries an implicit message. Our understanding of habitability has been shaped by surface life, by warmth and light and abundance. Antarctica represents a different axis of possibility, one defined by scarcity, stability, and time. This matters because it reframes how we think about unseen life. If organisms can thrive in these conditions, then environments we have dismissed as too extreme elsewhere on Earth may warrant closer attention. And within Antarctica itself, it suggests that the known examples are not isolated oddities, but hints of a broader biological landscape. There are still vast regions beneath the ice that have never been sampled. There are ice shelves whose undersides have never been observed. There are subglacial lakes known only from radar reflections. In each of these places, conditions resemble those where life has already been found. That does not guarantee that complex organisms exist everywhere beneath the ice. Science does not work that way. But it does mean that the question has shifted. It is no longer reasonable to ask whether life can exist in such places. The question now is what forms it takes, how diverse it is, and how long it has been there. As exploration continues, discoveries are likely to arrive quietly rather than dramatically. A new DNA sequence that does not match known organisms, a chemical signature that implies an unexpected metabolic pathway, a blurred shape on a camera feed that prompts further investigation rather than immediate explanation. These moments accumulate. They do not overturn understanding overnight. They refine it. Antarctica has become a catalogue of such refinements, each one expanding the map of life's possibilities. Creatures found where nothing should live are no longer anomalies. They are indicators, pointing toward deeper, still unseen systems operating beneath the ice. And as attention turns toward how isolation shapes those systems over millions of years, the story begins to shift from survival to transformation, from persistence to evolution, carried out quietly in the cold, far from any human witness. When life becomes isolated, truly isolated, cut off not just from predators or competitors, but from the wider flow of genes, seasons, and environmental upheaval, evolution does not stop. It slows, redirects, and begins to follow rules that feel unfamiliar to those of us accustomed to surface ecosystems. Antarctica's hidden environments offer some of the clearest natural examples of what evolution looks like when the outside world fades into irrelevance. Isolation beneath the Antarctic ice is not a metaphor. It is physical, chemical, and temporal. Kilometers of ice block movement, darkness removes visual cues, pressure seals water from the atmosphere. In many cases, these environments have remained closed for hundreds of thousands, even millions of years. No new species arrive casually. No migrations replenish genetic diversity. Whatever life exists, there must work with what it already has. This kind of isolation changes the very mechanics of evolution. On the surface, evolution is often driven by fluctuation. Climate shifts, food sources change, predators appear and disappear, populations rise and crash. Under these conditions, rapid adaptation can be advantageous. Flexibility matters, speed matters. Traits that allow quick responses to change are favoured. Beneath Antarctica's ice, the opposite is true. Conditions change very slowly. Temperature remains nearly constant, darkness is permanent. Nutrient availability fluctuates only within narrow bounds. In such environments, adaptability gives way to specialization. Traits that work well are preserved for long periods. Traits that waste energy are eliminated, not through dramatic competition, but through simple inefficiency. Evolution here is not about outpacing rivals, it is about minimizing loss. One of the most profound effects of isolation is genetic drift. In small populations, random changes in gene frequencies can have outsized effects. When populations are cut off and remain small for long periods, these random shifts accumulate. Over time, they can produce organisms that differ significantly from their ancestors, even in the absence of strong selective pressure. In Antarctica's hidden ecosystems, population sizes are likely limited by energy availability. There is only so much chemical fuel to go around. This constrains how many individuals can exist at any given time. Small populations mean that chance plays a larger role in shaping genetic outcomes. A mutation that would be insignificant in a large, interconnected population might become fixed simply because there are few alternatives. This process does not produce dramatic monsters or sudden leaps. It produces quiet divergence, subtle changes in enzymes, slight shifts in membrane composition, adjustments in reproductive timing or metabolic regulation. Over thousands of generations, these changes accumulate into distinct biological identities. Another consequence of isolation is the reduction of unnecessary traits. Maintaining complex structures costs energy. Eyes, pigmentation, rapid movement, elaborate behaviors all require resources. In environments where these traits provide little benefit, they tend to fade. This does not happen quickly, but over evolutionary timescales it is remarkably consistent. Many organisms in dark environments show some degree of sensory reduction. Eyes may become smaller, less complex, or disappear entirely. Pigmentation fades when camouflage and signaling are irrelevant. Musculature may be reduced when movement is infrequent. What remains are traits that directly support survival under constant conditions. This streamlining is not degeneration, it is optimization. In Antarctica's subglacial and sub-ice shelf environments, this optimization likely extends to molecular levels. Proteins must function reliably at low temperatures. Cell membranes must remain flexible despite cold and pressure. Metabolic pathways must extract maximum energy from limited resources. These requirements exert continuous subtle pressure on every aspect of cellular function. Over time, lineages that meet these demands persist. Those that do not disappear quietly. One of the most intriguing aspects of evolution in complete isolation is how it reshapes reproductive strategies. On the surface, many organisms rely on producing larger numbers of offspring, accepting that most will not survive. This strategy makes sense when environments are variable and mortality is high. In Antarctica's hidden worlds, producing many offspring would be wasteful. Resources are limited, conditions are stable. Survival depends on efficiency, not numbers. As a result, organisms are expected to invest more heavily in fewer offspring. Reproductive events may be rare. Development may be slow. In some cases, organisms may reproduce asexually, avoiding the costs of finding mates and producing specialized reproductive structures. These strategies further reinforce isolation. Asexual reproduction reduces genetic exchange even within populations. Sexual reproduction, if it occurs, may involve limited mate choice simply because few individuals exist. This creates feedback loops where divergence accelerates quietly. Time plays a critical role in all of this. Evolution in Antarctica's hidden environments unfolds over immense spans. A trait that takes a few thousand years to spread is not rushed. There are no seasonal deadlines, no sudden catastrophes forcing rapid change. This allows evolutionary pathways that would be unstable on the surface to persist here. It also means that some lineages may retain ancient characteristics lost elsewhere. Traits that disappeared in surface relatives due to competition or changing environments might remain functional here. In this sense, Antarctica may act as a biological archive, preserving evolutionary experiments from earlier eras. There is evidence for this idea in Antarctic marine life. Some species show close relationships to ancient lineages that once inhabited warmer oceans. As conditions cooled, these organisms adapted rather than retreating. Their surface relatives may have gone extinct or evolved into very different forms, while Antarctic populations continued along a quieter path. Isolation also affects how organisms interact. In small, stable communities, relationships between species can become highly specialized. Mutualistic interactions, where different organisms depend on each other, may become tightly integrated. Disrupting one partner could destabilize the entire system. Such interdependence increases vulnerability to change but enhances efficiency under stable conditions. It also means that entire ecosystems may represent unique configurations not found elsewhere. These systems are not interchangeable with surface ecosystems. They cannot be easily replaced if lost. Another subtle effect of isolation is the blurring of boundaries between species. In small populations with limited genetic diversity, distinctions can become less rigid. Some lineages may represent continua rather than discrete categories. From a human taxonomic perspective, this complicates classification. Organisms may not fit neatly into existing groups. This has already been observed in microbial communities beneath Antarctic ice. Genetic analyses reveal clusters of related organisms that do not correspond cleanly to known species. Some may represent ancient branches, others may be products of long isolation and drift. Understanding their relationships requires rethinking how diversity is defined. Isolation also shapes how organisms respond to stress. Surface life often encounters sudden challenges and evolves mechanisms to cope with rapid change. Beneath the ice, stress is constant but predictable. Organisms adapt to maintain stability rather than respond to shocks. This can result in highly robust systems within narrow parameters. As long as conditions remain within expected ranges, organisms function reliably. Outside those ranges, however, tolerance may be low. This makes isolated ecosystems particularly sensitive to external disturbance. From an evolutionary perspective, this sensitivity is a trade-off. Stability allows specialization, but specialization reduces flexibility. Antarctica's hidden life has likely accepted this trade-off over millions of years, betting on the persistence of its environment. This bet has paid off so far. Ice has come and gone, but large portions of the continent have remained isolated long enough for complex evolutionary stories to unfold unnoticed. Humans have arrived only recently, and even now, contact remains minimal. The most compelling implication of evolution in complete isolation is not that Antarctica harbours unknown creatures in the dramatic sense, but that it harbours life shaped by rules we rarely get to observe directly. These rules prioritize patience over speed, efficiency over expansion, endurance over dominance. Studying such systems challenges assumptions that come from surface-biased thinking. It forces a reconsideration of what successful evolution looks like. It suggests that complexity does not always arise from competition and change, but can also emerge from stability and constraint. As exploration continues, scientists are beginning to piece together evolutionary relationships between isolated Antarctic populations and their surface relatives. These comparisons offer glimpses into how life responds when stripped of familiar pressures. They also raise questions about how long such isolation can persist before external forces intervene. Climate change, ice shelf collapse, and increasing human presence threaten to disrupt these isolated systems. When isolation ends, evolution changes course. New species arrive, conditions fluctuate. Long-standing adaptations may become liabilities. Understanding evolution in complete isolation is therefore not just an academic exercise. It is a race against time to document systems that may soon experience their first significant disturbances in millions of years. And as attention turns next toward the deep sea beneath Antarctica's ice shelves, where isolation meets connection and ancient lineages encounter slow-moving currents from the wider ocean, the story becomes even more complex, blending solitude with subtle exchange in ways that continue to challenge our understanding of how life endures at the edges of possibility. The deep sea beneath Antarctica's ice shelves occupies a strange middle ground between isolation and connection. It is sealed off from sunlight and surface weather, yet it is not completely cut off from the rest of the ocean. It exists in shadow, shaped by ice above and rock below, influenced by currents that move so slowly they barely register as motion at all. For much of human history, this space was not just unexplored, but unimagined. The idea that anything significant might exist there felt unnecessary to consider. That assumption did not survive contact with evidence. Ice shelves form where glaciers flow outward from the continent and extend over the sea without immediately breaking apart. These floating platforms can be hundreds of meters thick, creating a ceiling that blocks light completely. Beneath them, seawater circulates in a dim, enclosed environment that remains at temperatures just below the freezing point. The pressure is immense, the darkness absolute, and the stability remarkable. Storms rage above, seasons pass. Below, very little changes. The depth beneath ice shelves varies. In some regions, the seafloor lies a few hundred meters below the ice. In others, it drops to depths exceeding a kilometer. The terrain is uneven, shaped by ancient geology and modified by slow sedimentation. Fine particles settle over time, carried in by currents or released from melting ice. This creates a soft seafloor in some areas, rocky outcrops in others, each offering different opportunities for life. One of the defining features of these environments is the scarcity of energy. Without sunlight, there is no local primary production in the traditional sense. Instead, food arrives indirectly. Organic particles produced in sunlit waters far away drift slowly beneath the ice shelves, sometimes taking months or years to reach the seafloor. By the time they arrive, they are sparse and degraded. Yet scarcity does not mean absence. Organisms living here are adapted to intercept what little energy becomes available. Filter feeders capture particles from the water column. Deposit feeders process sediments, extracting nutrients from material that has already passed through multiple biological hands. Scavengers take advantage of occasional larger inputs, such as the remains of animals that wander into these regions and die. This economy of reuse defines the deep sea beneath the ice. Matter circulates slowly, efficiently, and repeatedly. Nothing is wasted because nothing can be replaced easily. The first direct observations of these ecosystems were cautious and limited. Drilling through ice shelves is technically demanding and risky. Each borehole represents a narrow window into a vast hidden space. When cameras were finally lowered, expectations were low. The results were not dramatic in the way popular imagination might prefer, but they were profound. Life was there. Sponges anchored to rocks, filtering water with deliberate slowness. Starfish moving at a pace measured in hours rather than seconds. Worm-like organisms burrowing through sediment. Fish swimming through darkness with calm, unhurried movements. These were not transient visitors. They were residents, well adapted to the conditions. What struck researchers most was how ordinary these organisms seemed in form, yet how extraordinary their context was. Similar species exist elsewhere in the ocean, but here they live without light, under ice that has persisted for millions of years. Their presence suggested continuity rather than novelty, adaptation rather than reinvention. At the same time, genetic analyses began to reveal differences. Populations beneath ice shelves often show signs of long-term isolation. Gene flow with open ocean populations is limited. Currents beneath ice shelves are weak and directional. Entry points are few. Over time, this leads to divergence. Some organisms found beneath ice shelves may represent distinct species, even if they resemble known forms. Others may be part of species complexes, groups of closely related populations that differ subtly but significantly in genetics and physiology. These differences may reflect adaptations to constant cold, low energy availability, and stable conditions. One of the most intriguing questions concerns how larvae disperse in these environments. Many marine organisms rely on free-swimming larval stages to spread across large distances. Beneath ice shelves, this strategy becomes risky. Currents are slow, food is scarce, darkness is complete. Larvae drifting aimlessly may not survive long. As a result, some Antarctic species show modified reproductive strategies. Brooding, where parents protect developing offspring, is more common. Larval development may be shortened or altered. In some cases, larvae may be capable of feeding on unusual sources or surviving longer periods without food. These adaptations further reinforce isolation. When dispersal is limited, populations remain localized. Evolution proceeds independently in different regions beneath the ice. The underside of the ice shelf itself plays an active role in shaping these ecosystems. Melting and freezing at the ice-water interface create complex structures. Channels form where warmer water flows. Ridges develop where freezing dominates. These features influence water movement, nutrient distribution, and habitat availability. In some places, melting releases fresh water that alters local salinity and stratification. This can affect which organisms can survive there. In others, freezing concentrates salts, increasing density and causing water to sink, driving slow circulation patterns. These processes create a mosaic of micro-environments beneath the ice. Life responds to these variations. Some organisms cluster near areas of higher nutrient input. Others avoid zones of strong melting or freezing. Over time, these preferences shape community structure in ways that are difficult to predict without direct observation. There is also growing evidence that chemosynthesis plays a role in some sub-ice shelf ecosystems. Where seawater interacts with sediments rich in certain minerals, chemical reactions can produce energy sources usable by microbes. These microbes, in turn, may support higher organisms indirectly, either through direct consumption or by contributing organic matter to the system. If this is the case, then parts of the deep sea beneath ice shells function as hybrid systems, drawing energy both from distant photosynthesis and local chemistry. This combination could explain how relatively complex organisms persist in regions far from open water. What remains largely unknown is how widespread these processes are. Only a handful of sites have been studied in detail. Each has revealed unique characteristics. Extrapolating from such limited data is risky. Antarctica resists generalization. There are also indications that some deep-sea environments beneath ice shelves may be even more isolated than previously thought. In regions where ice shelves ground against underwater ridges, water exchange with the open ocean may be extremely limited. These pockets could remain stable for exceptionally long periods, allowing evolution to proceed with minimal external influence. Such environments would be prime candidates for harbouring organisms that differ markedly from their relatives elsewhere, not in size or appearance necessarily, but in physiology, metabolism, and life history. These differences may not be obvious at first glance. They require careful measurement, genetic analysis, and patience to uncover. The challenge is that exploration remains difficult. Each mission beneath an ice shelf requires significant resources. The risks are high. Equipment failures can end projects abruptly. Weather conditions limit access windows. Even when successful, the area explored is tiny relative to the whole. Despite these challenges, interest in these environments continues to grow. Not because of sensational possibilities, but because they offer rare opportunities to study life under conditions that have remained stable for geological timescales. They serve as natural laboratories for understanding long-term adaptation, resilience, and the limits of biological systems. They also carry implications beyond Earth. Ice-covered oceans are believed to exist on several moons in the outer solar system. The deep sea beneath Antarctic ice shelves provides one of the closest analogues available for studying how life might function in such places. The lessons learned here inform how scientists design missions, interpret data, and frame expectations elsewhere. At the same time, these ecosystems are not insulated from change. Warming oceans threaten to thin and destabilize ice shelves. As ice shelves retreat or collapse, environments that have been dark and stable for millions of years may suddenly be exposed to sunlight, warmer water, and new species. The consequences of such changes are uncertain. Some organisms may adapt, others may not. Ecosystems shaped by isolation may struggle to cope with rapid shifts. Documenting their current state is therefore urgent, not for preservation alone, but for understanding how life responds when long-standing boundaries are removed. The deep sea beneath the Antarctic ice shelves remains largely unseen, not because it is empty, but because it is difficult to reach and slow to reveal itself. Its inhabitants live without urgency, without light, without seasons, guided by constraints that have remained unchanged for longer than human history. As exploration continues, attention increasingly turns to how signals of life emerge indirectly in these environments, through chemistry, movement, and subtle changes that hint at organisms still beyond our direct observation, waiting not in hiding, but simply in the dark. In environments where direct observation is rare and fleeting, life often announces itself indirectly. Beneath Antarctica's ice, this indirect presence takes the form of signals rather than sightings, patterns rather than pictures, and clues that feel incomplete until placed alongside one another. Scientists working in these regions have learned to pay attention not only to what they can see, but to what instruments detect, to what chemistry implies, and to what refuses to behave as expected. One of the earliest and most persistent clues comes from chemistry. When researchers sample water from beneath ice sheets or ice shelves, they do not just look for organisms themselves. They analyse dissolved gases, isotopic ratios, nutrient concentrations, and byproducts of metabolic reactions. These chemical fingerprints can reveal biological activity even when organisms remain unseen. In several subglacial environments, measurements have shown unexpected concentrations of methane, hydrogen, and reduced iron compounds. On their own, these chemicals are not definitive proof of life. Geological processes can produce them as well. But when they appear alongside other indicators, such as specific isotopic signatures that tend to be associated with biological processing, the picture becomes more suggestive. For example, methane produced by microbes often carries a distinct isotopic pattern compared to methane produced by purely geological reactions. In some Antarctic subglacial samples, methane isotopes lean toward the biological end of the spectrum. This does not confirm the presence of particular organisms, but it strongly suggests that microbial communities are actively producing or consuming methane beneath the ice. Similarly, shifts in nitrogen compounds hint at ongoing biological cycling. Nitrogen is essential for life, but it rarely stays in one chemical form. Microbes convert it between states, such as nitrate, nitrite, and ammonia, depending on their metabolic needs. In water samples from beneath the ice, researchers have detected imbalances in these compounds that are difficult to explain without biological involvement. These chemical clues accumulate quietly. Each one on its own might be dismissed as ambiguous. Together they begin to outline metabolic networks operating out of sight. Another category of clues comes from particulate matter. When instruments detect organic particles suspended in subglacial or subice shelf water, questions arise about their origin. Some particles could be remnants of ancient material trapped when the ice formed. Others might arrive from distant open waters, carried slowly by currents, but in some cases the composition and freshness of the particles suggest local production. Fresh organic material breaks down over time. In cold environments, this process is slow, but it still occurs. When researchers find compounds that appear relatively unaltered, it implies recent synthesis. Without sunlight, recent synthesis points toward chemosynthetic organisms producing organic matter from inorganic sources. Then there are the genetic clues. Advances in molecular biology have allowed scientists to detect fragments of DNA and RNA in environmental samples, a technique known as environmental sequencing. This approach does not require capturing whole organisms. Instead, it identifies genetic material shed into the environment through cell death, reproduction, or metabolic activity. Environmental DNA recovered from Antarctic subglacial waters has revealed astonishing diversity. Many sequences match known groups of bacteria and archaea adapted to cold and low energy conditions. Others do not match any known organisms. These unknown sequences may represent entirely new species or deeply divergent lineages that have evolved in isolation. The presence of genetic material alone does not confirm active life. DNA can persist after death, especially in cold environments. However, the detection of RNA, which degrades quickly, provides stronger evidence of ongoing metabolic activity. In some Antarctic samples, RNA has been detected alongside DNA, indicating that organisms are not just present but active. Still, genetic clues raise as many questions as they answer. Without intact organisms, it is difficult to infer structure, behavior, or ecological role. A sequence might suggest a metabolic pathway, but not how it integrates into the broader ecosystem. It might hint at a novel enzyme, but not reveal what organism uses it or under what conditions. Beyond chemistry and genetics, there are physical signals that defy easy explanation. In some sub-ice shelf environments, instruments have detected subtle, repeated disturbances in water movement that do not align neatly with known current patterns. These disturbances are small, localized, and intermittent. They could be caused by melting or freezing processes at the ice interface. They could result from sediment shifts, or they could indicate biological activity, such as the movement of organisms large enough to displace water, but small enough to evade visual detection. Such signals are not dramatic. They do not resemble the sudden motion of a large animal passing by a camera. Instead, they appear as gentle fluctuations, persistent enough to notice but ambiguous enough to resist interpretation. Researchers are careful not to overstate their significance. At the same time, they do not ignore them. Another physical clue comes from acoustic data. Sound travels differently in cold, dense water beneath ice shelves. Instruments designed to measure ice movement or ocean circulation sometimes record unexplained acoustic signatures. These may be related to ice cracking, water flow, or interactions between ice and seafloor. In rare cases, patterns emerge that do not fit well with these explanations. Again, caution dominates interpretation. The Antarctic environment is complex, and many physical processes are still poorly understood. Assigning biological meaning to ambiguous signals risks crossing from science into speculation. Yet the persistence of unexplained patterns keeps attention focused. Shadows also play a role, both literally and metaphorically. When cameras are lowered into dark environments beneath ice shelves, visibility is limited. Artificial lights illuminate only a small area, and particles in the water scatter light unpredictably. In this context, shadows can be deceptive. A drifting particle close to the lens can appear larger than an organism farther away. Changes in lighting can create the illusion of movement. Nevertheless, there have been instances where cameras captured shapes that did not resolve clearly, forms that moved slowly at the edge of visibility, leaving behind uncertainty rather than confirmation. These moments are often excluded from public discussion because they do not meet standards of evidence. Within research teams, however, they prompt debate and further investigation. Such shadows are reminders of the limits of our tools. Cameras designed for deep-sea exploration struggle under ice shelves. Lighting must be minimal to avoid disturbing fragile environments. Navigation is constrained. Each frame captured represents a compromise between clarity and intrusion. In some cases, shadows are not visual at all, they are statistical. When models predict certain distributions of nutrients or chemical byproducts, and measurements consistently deviate from those predictions, something is influencing the system. That something may be biological, even if it remains unseen. For example, if oxygen levels drop slightly more than expected in a given region, it suggests consumption. If carbon dioxide levels rise subtly but persistently, it suggests respiration. When these patterns repeat across multiple samples and locations, they point toward active communities rather than isolated anomalies. Perhaps the most intriguing clues come from mismatches between expectation and observation. In several sub-ice shelf environments, researchers have found complex organisms living farther from open water than models predicted was possible. These organisms require a certain amount of energy to survive. Their presence implies either more efficient energy use than previously assumed, or additional energy sources not accounted for. These mismatches force revisions, currents are re-examined, sediment chemistry is reassessed, new hypotheses emerge, such as previously unrecognized chemosynthetic processes or long-range transport mechanisms that deliver organic matter more effectively than expected. Each revision expands the range of what might be possible. It is important to emphasize that none of these clues point toward large, undiscovered animals roaming beneath the ice in secret. The environment simply does not support that kind of biomass. Energy constraints alone rule out dramatic scenarios. What they do suggest is a diversity of life forms operating quietly, perhaps including organisms with body plans, behaviors, or metabolisms that differ from known examples. In particular, scientists remain interested in the possibility of soft-bodied organisms that leave little physical trace. Such organisms are notoriously difficult to detect even in well-studied environments. In Antarctica's hidden systems, they would be even more elusive. Their presence might be inferred only through chemical signals, genetic fragments, or indirect effects on surrounding communities. There is also the question of scale. Life beneath the ice does not need to be abundant to be significant. A small population persisting for millions of years represents a remarkable evolutionary achievement. Detecting such populations requires patience and sensitivity, not spectacle. This patience is reflected in how Antarctic research progresses. Data accumulates slowly. Hypotheses are refined cautiously. Years may pass between field seasons. Results are often incremental rather than revolutionary. Yet over time, a pattern emerges. That pattern suggests that Antarctica's hidden environments are biologically active, chemically dynamic, and more complex than early models predicted. It suggests that unseen life is not an anomaly, but an expected outcome of long-term stability, chemical energy availability, and isolation. The challenge lies in bridging the gap between signal and sight. Future exploration aims to do this through improved technology, autonomous underwater vehicles capable of navigating beneath ice shells for extended periods, sensors designed to detect biological activity more directly, sampling techniques that minimize contamination while maximizing information. Even with these advances, uncertainty will remain. Antarctica does not lend itself to complete understanding. There will always be regions beyond reach, processes beyond resolution, and life forms beyond classification. But the presence of signals, shadows, and strange biological clues serves as a reminder that absence of evidence is not evidence of absence. In the quiet beneath the ice, life leaves traces that reward careful attention. As researchers continue to follow these traces, the focus begins to shift toward why so much of Antarctica remains unexplored despite modern technology, and what barriers, both physical and conceptual, continue to limit our ability to see what lives there. Antarctica is often spoken about as if it has been explored, mapped, and largely understood, at least in broad strokes. Satellites orbit overhead. Research stations dot the coastline. Ice cores have been drilled. Maps exist. From a distance, it can feel as though the unknown has been reduced to manageable gaps. But this impression dissolves quickly when attention turns to life itself. Not the ice, not the climate history, not the physics of glaciers, but the living systems that exist on and beneath the continent. In that context, Antarctica remains one of the least observed biological regions on Earth. The reasons for this are not mysterious, but they are layered, reinforcing one another in ways that compound the difficulty. Some are logistical, some are technological, others are philosophical, shaped by how science prioritizes questions and defines feasibility. Together, they explain why so little of Antarctica's life has been seen, even as interest in the continent continues to grow. The most obvious barrier is access. Antarctica is remote in a way that resists casual travel. Reaching it requires long flights over open ocean or ice, dependent on weather windows that can close without warning. Once there, movement is slow and constrained. There are no roads crossing the interior. Aircraft land on ice runways that shift and crack. Vehicles must be carefully maintained to function in extreme cold. Even reaching a specific site can take weeks of preparation and coordination. This difficulty limits where research can occur. Most biological studies take place near established research stations, which are typically located along the coast. These areas are easier to supply and evacuate, and they offer access to relatively diverse ecosystems compared to the frozen interior. As a result, vast regions of Antarctica, particularly inland and beneath thick ice, remain untouched simply because getting there is too costly, risky, or impractical. Even when access is possible, time is limited. Antarctic field seasons are short. The austral summer offers a brief window of relatively stable conditions, but even then storms can halt work abruptly. Researchers may spend years planning a project only to have a single season cut short by weather. Missed opportunities are common. Long-term studies are difficult to maintain. Biological research suffers particularly under these constraints because life often requires repeated observation. Understanding ecosystems means tracking changes over time, monitoring populations and observing interactions. In Antarctica, such continuity is rare. Many studies rely on snapshots rather than sustained presence. This makes it harder to detect slow processes, rare organisms or subtle patterns. Technology presents another layer of limitation. Studying life beneath ice requires specialized equipment. Drilling through hundreds or thousands of meters of ice is expensive and technically demanding. Each borehole represents a significant investment. Once drilled, the opening is narrow, restricting what instruments can pass through and how they can move. Cameras lowered through these holes see only what falls within their limited field of view. Lighting must be carefully controlled to avoid disturbing environments or overwhelming sensors. Navigation is difficult. Remote vehicles risk becoming trapped beneath the ice, lost beyond recovery. For this reason, exploration is cautious and conservative. Sampling presents its own challenges. Collecting biological material without contaminating pristine environments requires strict protocols. Equipment must be sterilized. Fluids used in drilling must be carefully managed. The goal is to observe without altering, but achieving that balance is difficult. In many cases, researchers choose to limit sampling rather than risk a disturbance. This caution slows discovery, but it is necessary. Antarctica's isolated ecosystems may be particularly vulnerable to introduced organisms. Even a small contamination event could permanently alter systems that have remained stable for millions of years. Protecting these environments often means accepting limited data. Another reason so little has been seen is that Antarctic life does not announce itself in ways humans are accustomed to noticing. Much of it is microscopic. Even larger organisms tend to be slow, sparse, and well camouflaged. There are no dense forests or bustling reefs beneath the ice. Populations are thinly spread. Encounters are rare. In many cases, life exists below detection thresholds. Instruments designed to survey larger, more active ecosystems may simply miss it. This is especially true for soft-bodied organisms that do not leave obvious traces. Without shells or skeletons, they disappear quickly after death. Fossil records are poor. Visual detection is difficult. Bias also plays a role. Historically, Antarctic research focused on physical sciences, glaciology, meteorology and climate history took precedence. These fields offered clear global relevance and produced data that could be collected with relatively straightforward instruments. Biology, especially in hidden environments, received less attention. This focus shaped infrastructure and expertise. Research stations were designed to support ice drilling and atmospheric monitoring. Equipment and training emphasized those goals. Biological exploration beneath the ice remained a niche interest, pursued by small teams with limited resources. Conceptual assumptions reinforced this imbalance. For much of the 20th century, Antarctica was viewed as biologically marginal. Extreme cold and darkness were assumed to limit life severely. Where life existed, it was thought to be simple and sparse. This view discouraged exploration. Why look where nothing was expected? As evidence accumulated to challenge these assumptions, momentum began to shift. But scientific priorities changed slowly. Funding structures, institutional expertise, and established research programs are not easily redirected. Even today, biological exploration beneath Antarctica's ice competes with many other pressing scientific questions. Another factor is scale. Antarctica is enormous. Even if access were easy and technology unlimited, comprehensively surveying its biological systems would take decades. Beneath the ice sheet and ice shelves lies an area larger than many continents, much of it never directly observed. Sampling a few sites cannot capture this diversity. This scale amplifies uncertainty. When organisms are found in one location, it is difficult to know how representative that location is. Are similar communities widespread or isolated? Are observed species rare exceptions or common inhabitants? Answering these questions requires extensive coverage, which remains out of reach. Time also works against observation. Life in Antarctica often operates slowly. Growth rates are low. Reproductive cycles are long. Changes that might be obvious over months in temperate environments may take years or decades to detect here. Short-term studies can miss these dynamics entirely. In addition, many Antarctic ecosystems may experience long periods of apparent inactivity. Organisms enter dormant states when resources are scarce. Metabolism slows, movement ceases. During these periods, life is effectively invisible to many detection methods. A camera lowered at the wrong time might see nothing and conclude absence where there is only waiting. Human presence itself is another limiting factor. Antarctic research operates under strict environmental protections. Large-scale disturbance is prohibited. This limits the types of experiments that can be conducted. While this is ethically necessary, it constrains the scope of investigation. For example, extensive trawling or dredging beneath ice shelves is not acceptable. Large sample sizes cannot be collected casually. Researchers must rely on minimal intervention approaches, which yield less material but preserve integrity. This trade-off slows accumulation of biological knowledge. There is also the challenge of interpretation. When data are sparse and indirect, conclusions must be tentative. Scientists are trained to avoid overreach. As a result, many intriguing findings are framed conservatively, emphasizing uncertainty. This caution is appropriate, but it can give the impression that little is known, or that discoveries are less significant than they are. Public communication further complicates matters. Dramatic narratives attract attention, but Antarctic biology rarely offers drama in obvious forms. Discoveries are incremental. Significance lies in context rather than spectacle. As a result, much of what has been learned remains confined to academic literature, unnoticed outside specialist circles. All of these factors combine to create a situation where Antarctica's life remains largely unseen, not because it is absent, but because it is difficult to reach, slow to reveal itself, and easy to underestimate. Importantly, this situation is changing, though gradually. Advances in autonomous vehicles, remote sensing, and molecular biology are expanding what can be detected. International collaborations are pooling resources and expertise. Assumptions about habitability are being revised. Interest in extreme environments is growing, driven in part by astrobiology and the search for life beyond Earth. Still, progress will remain slow. Antarctica does not lend itself to rapid discovery. It demands patience, restraint, and a willingness to accept incomplete answers. As exploration continues, the question shifts from why we have seen so little to how we might responsibly see more. That shift brings with it new challenges, ethical considerations, and possibilities, particularly as attention turns toward the creatures that may exist beyond the limits of current observation, shaped by isolation and time in ways we're only beginning to imagine. When scientists talk about life that may exist but remains unnamed, they are not speaking in riddles or indulging in speculation for its own sake. They are describing a very specific gap between evidence and description, a space where data suggests presence, but classification has not yet caught up. In Antarctica's hidden environments, this gap is wide, persistent, and expected. It is not a failure of science. It is the natural consequence of limited access, extreme isolation, and biological systems that do not conform neatly to familiar categories. Naming a creature is not a casual act. It requires observation, repetition, comparison, and context. A species is not defined by a single encounter or a fragment of DNA. It must be described, differentiated, and placed within a broader framework of life. In environments where direct observation is rare and sampling is minimal, this process slows to a crawl. As a result, Antarctica is filled not only with unknown organisms, but with organisms that are known only partially, hinted at through chemistry, genetics, or indirect effects. One of the most common ways unnamed life announces itself is through genetic data. Environmental sequencing has revealed a wealth of DNA fragments in subglacial lakes, beneath ice shelves, and within ice itself. Many of these sequences resemble known groups of bacteria or archaea, but differ enough to resist precise identification. They sit at the edges of classification, close enough to suggest ancestry, distant enough to imply long independent evolution. In some cases, entire genetic lineages appear that have no close match in existing databases. These are not errors or contaminants. They recur across samples. They carry signatures of adaptation to cold, pressure, and low energy availability. They encode metabolic pathways that make sense in context, even if the organisms themselves have never been seen. Such lineages cannot be named properly without more information. A sequence alone does not reveal structure, behavior, or ecological role. It cannot show whether an organism is solitary or colonial, mobile or stationary, microscopic, or merely small. It tells part of a story, but not enough to assign a name with confidence. Beyond microbes, there is growing interest in the possibility of small, soft-bodied multicellular organisms that remain undocumented. These creatures, if they exist, would be particularly difficult to detect. Soft tissues decay quickly. Without shells, bones, or other durable structures, they leave little trace behind. In energy-poor environments, their populations would likely be sparse, making encounters rare. In the deep sea beneath ice shelves, Similar organisms elsewhere on Earth are often overlooked even with extensive sampling. In Antarctica's hidden systems, where sampling is minimal and visual surveys are limited, their absence from records does not imply non-existence. It implies invisibility. The environments themselves encourage this invisibility. Darkness eliminates visual signaling. Cold slows movement. Stability removes the need for dramatic behaviours. An organism that spends most of its life attached to a surface, feeding slowly on chemical gradients or suspended particles, may never cross a camera's field of view during a brief observation window. Some unnamed creatures may not even resemble what we think of as animals. They may blur boundaries between categories. Colonial organisms composed of repeated units. Symbiotic assemblies where multiple species function as a single ecological entity. Life forms whose individuality is difficult to define. Such complexity challenges taxonomy, which relies on clear distinctions. In isolated environments, those distinctions may soften. Evolution under stable conditions can favour integration rather than diversification. Organisms may become more interdependent, their identities intertwined. Another source of unnamed life comes from metabolic novelty. In several Antarctic subglacial environments, chemical data suggest reactions that do not align perfectly with known metabolic pathways. These anomalies are subtle. Slight imbalances in expected ratios, unexpected byproducts, energy yields that do not fit established models. One explanation is that known organisms are behaving in unfamiliar ways under extreme conditions. Another is that unknown organisms are present, employing pathways that have not yet been described. Without isolating and studying these organisms directly, it is impossible to say which explanation is correct. This uncertainty does not remain static. As models improve and datasets grow, some anomalies are resolved, others persist. Persistent anomalies are often the most interesting, not because they promise sensational discoveries, but because they resist simplification. In Antarctica, persistence is a theme. Consider the possibility of organisms adapted to exploit extremely low energy gradients. In most ecosystems, energy differences are relatively large. Light versus dark, hot versus cold, rich versus poor. Beneath the ice, gradients are faint. A slight difference in mineral concentration, a small temperature variation over distance. Exploiting such gradients requires extraordinary sensitivity and efficiency. Organisms capable of doing this might operate at metabolic rates so low they approach detection limits. They might grow imperceptibly. Their activity might register only as subtle chemical shifts over long periods. From a human perspective, they would appear almost inert, yet inert does not mean absent. There is also the question of scale. Some unnamed creatures may be larger than microbes, but smaller than typical animals, occupying a middle ground that receives little attention. Meiofauna, organisms that live between sediment grains, often fall into this category. In temperate oceans, they are abundant and diverse, yet underrepresented in studies because they require specialized sampling and microscopy. In Antarctic sub-ice environments, meiofauna could exist in sediments beneath ice shelves or in subglacial lakes. Their presence would be easy to miss without targeted effort. Their classification would be challenging due to potential divergence from known forms. Then there are organisms that exist primarily in dormant states. Some life forms may spend the majority of their existence in suspended animation, becoming active only under rare conditions. In Antarctica, such conditions might arise from episodic nutrient pulses, minor shifts in water flow, or geological activity. If sampling does not coincide with these periods of activity, these organisms remain effectively invisible. Their DNA may be present, but their behaviour and form remain unknown. Naming such creatures requires catching them in motion, not merely detecting their genetic shadow. Another complicating factor is the possibility of ancient lineages that have changed little over time. In stable environments, strong selective pressure for change may be absent. Organisms that function adequately persist without modification. Over millions of years, they become living relics, not because they are primitive, but because they are well suited to their niche. Such lineages may retain traits that disappeared elsewhere. Their genetic signatures may appear unusual when compared to modern surface organisms. They may be misclassified or dismissed as odd variants, rather than recognized as distinct entities. Recognition requires context, and context is sparse. There is also the matter of symbiosis. In extreme environments, cooperation can be more advantageous than competition. Organisms may form tight partnerships, exchanging nutrients or metabolic products. In some cases, one organism may live entirely within another, blurring the line between host and symbiont. If such systems exist beneath Antarctica's ice, they complicate naming conventions. Is the unit of interest the host, the symbiont, or the combination? Traditional taxonomy struggles with such questions, even in well-studied environments. Unnamed creatures are not necessarily rare. They may be common within their niche, simply overlooked. Their anonymity reflects our limited perspective rather than their insignificance. Importantly, scientists do not approach these possibilities with unchecked imagination. They work within constraints imposed by energy availability, chemistry, and physics. Antarctica does not support large, fast-moving predators or dense populations of complex animals beneath its ice. The unnamed creatures likely to exist there are modest in scale, subtle in behaviour, and efficient in design. Their significance lies not in their size or drama, but in what they reveal about life's flexibility. The process of naming such creatures will be slow. It will involve incremental advances in sampling, imaging, and analysis. It will require patience and restraint. Many candidates will remain unnamed for decades, known only through indirect evidence. This is not unusual in science. Deep-sea biology offers parallels. Entire ecosystems were inferred from chemical anomalies before being observed directly. Hydrothermal vent communities were suspected long before cameras captured them. In each case, naming followed observation, not anticipation. Antarctica's hidden life may follow a similar trajectory, though stretched over longer timescales due to access limitations. There is also an ethical dimension. Naming a species implies a certain claim, a recognition that brings attention and potentially exploitation. In Antarctica, where ecosystems are fragile and protected, scientists must balance curiosity with caution. Some argue that leaving organisms unnamed until they can be studied responsibly is preferable to rushing classification. This restraint aligns with a broader shift in how exploration is conducted. The goal is not to catalogue everything as quickly as possible, but to understand systems without destabilizing them. As technology improves, the list of unnamed creatures will likely grow before it shrinks. Better detection reveals more complexity. More complexity reveals more gaps. Each gap represents an opportunity for discovery, but also a reminder of how little has been seen. These unnamed creatures occupy a conceptual space that is easy to misunderstand. They are not myths waiting to be confirmed or denied. They are hypotheses grounded in data, shaped by constraints, and refined through careful analysis. They represent the frontier not of imagination, but of evidence. In Antarctica, that frontier is vast. As exploration continues, attention increasingly turns toward how ancient some of these lineages may be, and whether the ice has preserved not just isolated populations, but entire evolutionary histories that have unfolded quietly beneath the frozen surface. When scientists speak about ancient lineages frozen in time, they are not suggesting that life beneath Antarctica's ice has stopped evolving. Evolution never truly stops. What they mean is something subtler and far more interesting. In environments where conditions remain stable for immense spans of time, where disturbance is rare and resources change only gradually, evolution can proceed so slowly and consistently that entire lineages retain recognizable characteristics for millions of years. Change still occurs, but it accumulates at a pace that makes the present feel closely connected to the deep past. Antarctica is uniquely suited to preserving such lineages. Large portions of the continent have remained isolated since long before humans existed, long before many modern ecosystems took shape. While surface environments elsewhere were repeatedly reshaped by ice ages, warming periods, rising seas and mass extinctions, Antarctica's buried and sub-ice environments experienced a different kind of history. They were sealed, buffered, and insulated from the most violent swings of planetary change. This insulation does not make Antarctica timeless, but it does make it selective about what changes matter. To understand how ancient lineages persist, it helps to consider what typically drives extinction and rapid evolution. Sudden climate shifts, competition from invasive species, habitat loss, and catastrophic events all impose strong selective pressures. Species that cannot adapt quickly disappear. Those that can may change dramatically over relatively short periods. On the surface, these pressures are frequent and intense. Beneath Antarctica's ice, many of these drivers are absent or muted. There are no invasive species arriving casually. There are no seasonal collapses of food webs. Temperature changes are gradual. Habitat structure remains consistent over long spans. Under these conditions, species that are well adapted have little reason to change. This creates an environment where evolutionary conservatism can be advantageous. Traits that work continue to work. There is no reward for novelty if novelty consumes energy without providing benefit. Over time, this favours organisms that are stable in form and function. Evidence for this kind of persistence already exists in Antarctic marine life close to the surface. Some Antarctic invertebrates show clear links to ancient groups that once dominated warmer oceans. Their relatives elsewhere have diversified or disappeared, while Antarctic populations retained ancestral traits, modified just enough to function in colder conditions. These organisms are not fossils in the literal sense. They are living, reproducing species, but their basic body plans, developmental pathways, and ecological roles resemble those of their distant ancestors more closely than those of many modern counterparts. In deeper, more isolated environments beneath ice shelves and ice sheets, the potential for such preservation increases. Isolation limits competition. Stability reduces pressure for rapid change. Small population sizes encourage genetic drift, but drift alone does not necessarily produce dramatic morphological shifts. Instead, it often fine-tunes existing traits. Over millions of years, this combination can result in lineages that appear strikingly old in genetic terms, even if they remain functionally competent. Genetic studies hint at this possibility. Some DNA sequences recovered from subglacial environments show deep divergence from known lineages. They do not cluster closely with modern groups. Instead, they branch off earlier, suggesting long independent histories. These sequences are not relics from dead organisms. They appear alongside evidence of active metabolism, indicating living populations. Interpreting such data is challenging. Without whole organisms, it is difficult to reconstruct evolutionary relationships with confidence, but patterns emerge. Some lineages appear to have split from known groups tens of millions of years ago. That timing aligns with major climatic shifts that isolated Antarctica and reshaped global circulation. When Antarctica separated from other continents and circumpolar currents formed, the continent cooled dramatically. Ice advanced. Marine and terrestrial environments were transformed. Many species retreated northward or went extinct. Others remained and adapted. Those that remained entered a long period of isolation. Isolation does not guarantee preservation, but it makes it possible. In subglacial lakes, for example, water bodies may have been sealed beneath ice for hundreds of thousands or even millions of years. If life existed there before sealing, it would have been trapped. Subsequent evolution would proceed without outside influence. Over such timescales, even microbes could develop unique traits that reflect ancient conditions. These traits might include metabolic pathways that were more common in earlier Earth environments but are rare today. Early Earth chemistry differed from modern conditions. Oxygen levels were lower. Certain elements were more available. Microbes that evolved under those conditions developed strategies that later became less advantageous elsewhere. Antarctica's hidden environments may preserve echoes of those strategies, not because they are ancient in origin necessarily, but because they remain useful under current conditions there. This possibility fascinates researchers because it offers a glimpse into evolutionary pathways that were largely overwritten elsewhere. Studying such organisms could reveal how life functioned under earlier planetary conditions and how it adapts when modern pressures are absent. There is also the possibility that some lineages beneath the ice originated after isolation, but followed evolutionary trajectories that resemble ancient patterns due to similar constraints. Evolution does not have memory, but it does respond to constraints in repeatable ways. Limited energy, stable environments, and isolation can produce outcomes that resemble early life strategies even in relatively recent lineages. Distinguishing between genuinely ancient ancestry and convergent evolution is difficult. It requires extensive genetic comparison, biochemical analysis, and ecological context. In Antarctica, where data are sparse, these distinctions remain provisional. Another factor contributing to the persistence of ancient lineages is low extinction pressure. On the surface, species often go extinct because they are outcompeted, preyed upon, or unable to cope with change. Beneath Antarctica's ice, competition is limited. Predator-prey relationships exist, but at low intensity. Extinction still occurs, but it is driven more by gradual inefficiency than by sudden collapse. This slower turnover allows lineages to persist longer. Species that might be displaced elsewhere by faster-growing competitors can continue to exist here because no such competitors arrive. Stability becomes a shield. However, this shield has limits. Geological events still occur. Ice sheets advance and retreat. Water networks shift. Over very long timescales, environments can change enough to alter selection pressures. But compared to most of Earth's surface, these changes are modest. The idea of lineages frozen in time also intersects with how humans perceive change. Our sense of history is short. A species that remains relatively unchanged for a few million years feels ancient to us, even though it has experienced countless generations. From an evolutionary perspective, this is not stasis, but steady continuity. This continuity is particularly compelling in Antarctica because it is paired with physical preservation. Ice traps atmospheric gases, sediment preserves chemical signatures, and water bodies remain sealed. Biological and geological archives coexist. This allows researchers to correlate biological data with environmental history in ways that are difficult elsewhere. For example, changes in ice chemistry might align with shifts in genetic markers, suggesting periods of stress or adaptation. Sediment layers might record nutrient availability changes that correspond to evolutionary adjustments in resident organisms. These correlations are still largely theoretical, but they represent a powerful research direction. Ancient lineages also raise questions about vulnerability. Organisms adapted to long-term stability may struggle when conditions change rapidly. As climate warming affects Antarctica, ice shelves thin, meltwater increases, and circulation patterns shift. Environments that were once stable may become dynamic. Lineages that persisted for millions of years could face unprecedented challenges within centuries. Their specialized adaptations may become liabilities. Understanding their biology now is crucial to predicting their fate. There is also a philosophical aspect to this discussion. Humans tend to equate progress with change. Evolutionary success is often framed as rapid diversification or dominance. Ancient lineages challenge that narrative. They demonstrate that persistence itself can be a form of success. These organisms have survived not by outcompeting others, but by fitting so well into their niche that change was unnecessary. Their existence is a reminder that evolution does not aim toward complexity or novelty. It aims towards sufficiency. In Antarctica's hidden worlds, sufficiency takes on a particular form: energy efficiency, structural simplicity, longevity. These traits align with what scientists observe in many ancient lineages elsewhere, such as certain deep-sea organisms or extremophils in isolated environments. What makes Antarctica unique is the scale and continuity of isolation. Few places on Earth offer such extensive, long-term separation from surface processes. This makes the continent a natural laboratory for studying how ancient evolutionary patterns persist under modern conditions. As exploration continues, researchers hope to identify clearer links between Antarctic organisms and ancient lineages. This may involve comparing genetic data across continents, reconstructing ancestral states, and integrating geological history with biological findings. Each step forward is slow, samples are limited, interpretation is cautious, but the possibility remains compelling. Beneath the ice, life may not just survive, it may remember. And as attention shifts toward what these ancient lineages can teach us about life beyond Earth, particularly in similarly isolated and stable environments elsewhere in the solar system, Antarctica's role in the broader story of biology becomes even more significant, connecting the deep past of our planet with questions about life's potential future far beyond it. When scientists look to Antarctica for lessons about life elsewhere, they are not engaging in metaphor or speculation for its own sake. They are responding to a simple and increasingly well-supported idea. If life can persist beneath kilometers of ice, in darkness, under pressure, with minimal energy input, then the boundaries we place on habitability may be far narrower than reality demands. Antarctica does not just challenge what we think life needs, it quietly expands the list of places where life could exist. For much of scientific history, the search for life beyond Earth focused on familiar conditions: liquid water at the surface, moderate temperatures, sunlight, a stable atmosphere. These criteria were not arbitrary. They were based on the only example of life we knew. Earth's surface biosphere shaped expectations, models, and mission designs. Planets were judged by how closely they resembled our own, and those that did not were quickly dismissed. Antarctica complicates this perspective. Here is a place on Earth where life operates almost entirely outside the familiar framework. Sunlight is irrelevant in many environments. Surface conditions are hostile to complex organisms. Energy arrives through chemistry and slow transport rather than direct production. And yet, ecosystems function. They persist. They adapt. They endure for geological timescales. This forces a reconsideration of what it means for a world to be habitable. One of the most important lessons Antarctica offers is that liquid water does not need to be obvious or abundant at the surface to support life. Beneath Antarctic ice sheets and ice shells, water persists due to pressure, salinity, and geothermal heat. It forms networks of lakes, channels, and marine environments that remain hidden from view. These water bodies are stable, insulated, and long-lived. This has direct implications for worlds beyond Earth. Several moons in the outer solar system are believed to harbour subsurface oceans beneath thick ice shells. These oceans are not speculative. Multiple lines of evidence suggest their existence. Gravity measurements, magnetic field interactions, surface geology, and plume observations all point toward liquid water hidden below ice. For a long time, the question was whether such oceans could support life. Antarctica provides a partial answer. Life does not require sunlight if alternative energy sources are available. Chemosynthesis can sustain ecosystems, as it does near deep-sea vents and beneath Antarctic ice. Chemical gradients, water-rock interactions, and geothermal energy can all provide usable energy. The key is not abundance, but continuity. Antarctica demonstrates that continuity can exist in isolated environments for millions of years. Another lesson concerns stability. Surface environments on Earth are dynamic. Climate shifts, seasons change, ecosystems rise and fall. Beneath Antarctica's ice, conditions are far more stable. Temperature varies little. Darkness is constant. Nutrient availability changes slowly. This stability allows life to adapt deeply rather than quickly. In astrobiology, stability is often overlooked in favour of surface habitability. Yet stability may be just as important. A world that is harsh but consistent may be more hospitable to life over long time scales than one that fluctuates wildly. Antarctica shows how life can specialize for narrow conditions and persist without needing to cope with constant upheaval. This specialization does not produce fragility in the way one might expect. Antarctic organisms are not delicate. They are resilient within their niche. Their vulnerability lies not in their environment, but in sudden change. This distinction matters when considering extraterrestrial environments. Worlds that appear hostile by Earth standards may be perfectly suitable for life adapted to their specific conditions. Antarctica teaches that judging habitability by surface comfort is misleading. Another important lesson comes from scale. Antarctica's hidden ecosystems operate at low energy levels. Biomass is limited. Populations are sparse. Growth is slow. From a distance, such systems might appear lifeless. Traditional detection methods that look for large-scale biological signatures could miss them entirely. This has implications for how we search for life elsewhere. If life exists on other worlds in low-energy forms, it may not produce strong atmospheric signatures or obvious surface features. It may not alter its environment dramatically. Detecting it will require sensitivity, patience, and an openness to subtle clues. Antarctica reminds us that life does not always announce itself loudly. The continent also teaches humility about assumptions. Before subglacial lakes were discovered, few scientists expected liquid water beneath kilometers of ice. Before life was found there, even fewer expected active ecosystems. Each discovery forced a revision of models and expectations. This pattern is instructive. When exploring other worlds, we should expect surprises. Models based on limited data will be incomplete. Antarctica shows how easily entire categories of environments can be overlooked until technology improves or perspectives shift. Another lesson concerns isolation. Antarctica's hidden environments are isolated not just spatially, but biologically. Gene flow is limited. Migration is rare. Evolution proceeds independently. This produces unique lineages shaped by local conditions rather than global competition. In the context of life elsewhere, isolation may be the norm rather than the exception. Subsurface oceans, underground aquifers, or ice-covered habitats on other worlds would likely be isolated from one another. Life in such places would evolve locally, producing diversity not through interaction, but through separation. This suggests that if life exists elsewhere, it may be highly localized. Different environments on the same world could host entirely different forms of life, each adapted to its niche. Antarctica offers a preview of how such diversity might arise quietly and remain hidden. Antarctica also challenges ideas about complexity. It is tempting to equate complexity with visibility or size. Yet some of the most sophisticated adaptations in Antarctic organisms occur at molecular and biochemical levels. Antifreeze proteins, cold-adapted enzymes, efficient metabolic pathways, and robust repair mechanisms are forms of complexity that do not manifest as elaborate structures. When searching for life elsewhere, focusing solely on macroscopic complexity may be misleading. Life may be complex in ways that require detailed analysis to appreciate. Antarctica teaches that sophistication does not always scale up to spectacle. There is also an important lesson about time. Life beneath Antarctica's ice has had millions of years to adapt to its environment. It did not need to arise quickly or spread rapidly. It needed only to persist. Given enough time, even marginal environments can support stable ecosystems. This has implications for planetary history. Worlds that were once more hospitable may still host life in refuges, long after surface conditions became hostile. Antarctica itself underwent dramatic cooling, yet life adapted and persisted beneath the ice. This suggests that life elsewhere could survive planetary changes by retreating into protected environments. The idea of refugees is central here. Antarctica shows how ice can act not just as a barrier, but as protection. Ice insulates, stabilizes, and preserves. It blocks radiation. It dampens temperature swings. It isolates ecosystems from surface catastrophes. In this sense, ice-covered worlds may be more hospitable than they appear. Rather than killing life, ice may shelter it. Another lesson comes from how difficult it is to observe these systems. Antarctica's hidden life was not discovered because it was obvious. It was discovered because scientists asked questions that went against prevailing assumptions. They drilled where no one expected water. They sampled where nothing was predicted to live. This suggests that the search for life elsewhere must be similarly willing to challenge assumptions. Missions designed only to confirm existing models may miss what lies outside them. Antarctica rewards curiosity that is patient and open-ended. There is also an ethical lesson embedded here. Antarctica's ecosystems are fragile precisely because they have been stable for so long. Disturbance can have lasting consequences, As we consider exploring other worlds, especially those that may host life, Antarctica reminds us to proceed carefully. Exploration is not neutral, it changes what it touches. The goal should not be conquest or extraction, but understanding. Finally, Antarctica teaches that life is not defined by comfort or familiarity. It is defined by persistence, by the ability to make use of available energy, by adaptation to constraint rather than abundance. This reframing matters deeply for how we imagine life beyond Earth. Instead of asking whether other worlds look like ours, we might ask whether they offer stability, energy gradients, and time. If they do, life may find a way. Antarctica stands as proof that even on our own planet, entire biospheres can exist beyond ordinary perception, thriving quietly under conditions once thought impossible. It suggests that the universe may be more biologically generous than we have assumed, not because it offers many Earth-like worlds, but because life itself is more flexible than we imagined. As research continues, Antarctica remains a bridge between the known and the unknown, a place where lessons about life's resilience are written not in abundance or spectacle, but in patience and endurance. And as attention turns next toward the ethical questions raised by discovery about whether all hidden worlds should be explored or disturbed, Antarctica's example becomes not just scientific, but philosophical, forcing us to consider not only what we can find, but what we should leave unseen. Discovery is often spoken about as an unquestioned good, to find, to see, to know. These ideas sit comfortably at the heart of science and exploration. They suggest progress, understanding, and enlightenment. But in places like Antarctica, especially in its hidden biological worlds beneath ice and ocean, discovery carries a weight that cannot be ignored. Every act of observation is also an act of intrusion, and every new piece of knowledge arrives with consequences that extend beyond curiosity. Antarctica is not empty in the moral sense. It is not a blank slate waiting for human attention. It is a place that has existed largely without us, shaped by processes that do not care whether they are observed or named. Its ecosystems, particularly those beneath the ice, have persisted in isolation for spans of time that dwarf human history. That isolation is not incidental. It is fundamental to how these systems function. The ethical challenge begins with a simple question. When we are capable of reaching such environments, should we? Scientific exploration has always involved disturbance. Digging, sampling, collecting, measuring. On most of Earth, ecosystems are already shaped by human activity. The marginal impact of one more study is often small compared to existing pressures. Antarctica is different. Many of its hidden ecosystems may never have encountered complex life from the surface, let alone humans. Introducing foreign material, even accidentally, risks altering systems that evolved without such influence. This is not a hypothetical concern. Microorganisms are resilient. They travel easily. A single bacterium introduced into a subglacial lake could, in theory, survive and proliferate. Even if it did not outcompete native organisms, its presence would change the system. Chemical balances would shift. Interactions would alter. A record of millions of years of isolation would be permanently compromised. This is why Antarctic research operates under some of the strictest environmental protocols in the world. Drilling fluids must be sterilized. Equipment is cleaned repeatedly. Sampling is minimized. In some cases, scientists choose not to sample at all, relying instead on indirect measurements. Knowledge is sacrificed deliberately to protect integrity. This restraint is unusual in the history of exploration. It represents a shift away from the idea that anything reachable should be examined directly. Instead, Antarctica demands a more cautious posture, one that treats ignorance as sometimes preferable to irreversible change. But restraint has limits. Without sampling, understanding remains incomplete. Indirect data can suggest life, but it cannot fully describe it. Without organisms to study, we cannot learn how they function, how they evolved, or how they respond to change. In the context of a warming planet, this ignorance carries its own risks. If ecosystems are changing rapidly, failing to document them now could mean losing them without ever understanding what they were. This creates a tension that has no simple resolution. On one side lies the risk of disturbance, on the other lies the risk of permanent ignorance. The ethical landscape becomes even more complex when considering climate change. Antarctica is not static anymore. Ice shelves are thinning, meltwater is increasing. Environments that were once sealed are being exposed naturally, without human intervention. In some cases, sunlight is reaching places that have been dark for millions of years. Ocean circulation patterns are shifting. New species are arriving from warmer regions. These changes raise difficult questions. If an ecosystem is going to be altered by climate forces regardless, does that change the ethics of studying it? Does urgency justify greater intervention? Or does it demand even more caution, given the fragility of systems already under stress? There is no consensus answer. Some scientists argue that accelerated study is essential. They point out that understanding how these ecosystems function could inform conservation, climate modelling, and even global biological theory. Waiting too long could mean missing critical information about life's limits and resilience. Others argue that increased intervention, even for research, risks compounding harm. They emphasize that Antarctica's value lies partly in its role as a control, a place where natural processes unfold with minimal human influence. Disturbing it further, even in the name of knowledge, undermines that role. This debate extends beyond Antarctica itself. The continent has become a testing ground for how humanity approaches untouched environments. The decisions made here set precedents for future exploration, including the exploration of other worlds. If life exists beneath the ice of distant moons or within subsurface oceans elsewhere, the ethical questions will be similar. Should we drill into those environments? Should we introduce instruments, probes, or samples that could contaminate them? Or should we limit ourselves to remote observation, accepting uncertainty? Antarctica offers a rehearsal for these decisions. Another ethical dimension involves naming and ownership. Naming a species is a powerful act. It places that organism within a human framework. It asserts a kind of recognition, but also a kind of claim. In Antarctica, where no nation owns the land and no individual can assert sovereignty, naming still carries symbolic weight. Some argue that naming organisms found in such isolated environments risks encouraging a sense of possession. Once something is named, it becomes easier to justify studying it intensively, manipulating it, or even exploiting it in the future. Others argue that naming is a form of respect, a way of acknowledging existence rather than ignoring it. This tension reflects broader questions about how humans relate to the natural world. Are we observers, participants, or stewards? Does understanding imply responsibility, or does it create entitlement? Antarctica complicates these questions because it resists human presence so thoroughly. There is no illusion of harmony here. Humans do not belong easily. Every footprint is temporary. Every structure must be maintained constantly against the environment. This resistance encourages humility, but it does not enforce it. Ethical exploration requires conscious choice. There is also the issue of scale. Individual research projects may be small, but collectively they add up. Each borehole, each sample, each instrument contributes to cumulative impact. Coordinating research to minimize overlap and redundancy is an ethical challenge as much as a logistical one. International cooperation plays a critical role here. Antarctica is governed by treaties that prioritize peace and science. These agreements limit commercial exploitation and encourage shared stewardship. They also require consensus, which slows decision-making but helps prevent unilateral actions driven by narrow interests. Still, pressures exist. Scientific competition, funding cycles, and national prestige can push toward more aggressive exploration. Balancing these pressures against environmental responsibility requires constant vigilance. Another ethical consideration involves uncertainty itself. Many of the environments beneath Antarctica's ice are poorly understood. Acting without full knowledge increases the risk of unintended consequences. Ethical frameworks often emphasize informed consent, but ecosystems cannot consent. Their protection relies on human judgment, which is necessarily imperfect. This places a heavy burden on researchers. Decisions must be made with incomplete information, balancing potential benefits against unknown risks. In such cases, caution often becomes the default. But caution can also delay understanding that might ultimately aid protection. The ethics of disturbance also intersect with public perception. Dramatic narratives about hidden life can attract attention and support for research, but they can also encourage sensationalism. Overstating discoveries or implying mystery where evidence is thin can distort priorities and undermine trust. Responsible communication is part of ethical exploration. Presenting findings accurately, emphasizing uncertainty, and avoiding exaggeration help ensure that decisions are guided by understanding rather than excitement. At its core, the ethical challenge of Antarctica is not about choosing between knowledge and ignorance. It is about choosing how to pursue knowledge in a way that respects what already exists. Some environments may be best left largely untouched, studied indirectly, their integrity preserved even at the cost of detailed understanding. Others may warrant careful, limited sampling to answer pressing questions. These decisions cannot be standardized. They must be made case by case, informed by science, ethics, and humility. Antarctica forces these considerations into the open because it offers no easy justifications. There are no economic imperatives that clearly outweigh environmental concerns. There are no resident human populations whose livelihoods depend on exploitation. What remains is curiosity, responsibility, and choice. In this sense, Antarctica is not just a place of scientific inquiry. It is a mirror. It reflects how humanity approaches the unknown, how it balances desire with restraint, and how it defines progress. As exploration continues, the continent will continue to challenge us not only with what it hides, but with how we choose to reveal it. And as we move toward the final reflections of this journey, the focus turns from what may exist beneath the ice to what our relationship with such unseen worlds says about us and about the kind of explorers we choose to be. It is tempting at the end of a long exploration to imagine ourselves standing above it all, to picture Antarctica as a place we have finally looked into, understood a little better, reduced from mystery to knowledge. But that instinct does not sit comfortably here. Antarctica does not feel like a place that yields. It feels like a place that tolerates observation, briefly, selectively, without altering its own priorities. When we think about the creatures beneath the ice, the ones we have seen and the ones we only suspect, it is easy to frame the relationship in one direction. We observe, we measure, we infer, we imagine life hidden below, unaware of us, continuing on its slow, insulated path regardless of our curiosity. Yet there is another way to think about this exchange, one that feels more fitting to the continent itself. Antarctica does not invite attention. It does not signal abundance or demand understanding. It remains quiet, vast, and largely indifferent to human presence. In that quiet, the life that exists there has adapted not only to cold and darkness, but to absence. Absence of disturbance, absence of novelty, absence of observers. From that perspective, our arrival is the anomaly. Every drill hole, every lowered camera, every sensor introduced beneath the ice represents a momentary interruption in systems that evolved without interruption as a defining feature. The organisms living there did not evolve to evade predators like us, or to attract our interest. They evolved to persist, to occupy narrow margins of energy and time with remarkable efficiency. In that sense, it is not unreasonable to imagine that the relationship is not purely observational, not because these creatures are aware of us in any conscious or intentional way, but because our presence introduces change, however slight. Temperature shifts, chemical traces, vibrations, light in places where light has never existed. Life responds to change. That is what it does. A microbial community does not need awareness to register disturbance. It only needs chemistry. An organism attached to the seafloor does not need curiosity to be affected by altered currents or introduced particles. The systems beneath the ice are sensitive precisely because they are stable. They notice deviations not through perception, but through response. This is where the idea of being watched back takes on a different meaning. It is not about eyes in the dark or unknown observers tracking our movements. It is about feedback, about interaction, about the fact that even passive observation alters the system being observed. Antarctica makes this visible in a way few places do because the contrast between before and after is so stark. Before humans arrived, these environments changed slowly, governed by geological time and chemical balance. After arrival, even minimal interference introduces a new variable. Life beneath the ice adjusts, not because it recognizes us, but because it must. This realization carries weight. It reframes discovery as participation rather than distance. We're not outside these systems. The moment we reach them, we become part of their story. That story is not dramatic. It does not involve confrontation or revelation. It unfolds quietly, at the pace of Antarctic life itself. A slight shift here, a gradual response there. Effects that may not be noticeable for years or decades. And yet, that quiet interaction is precisely what makes Antarctica so powerful as a place of reflection. The creatures beneath the ice, named or unnamed, ancient or newly adapted, do not exist to be found. They exist because conditions allowed them to endure. Their success is measured not in expansion or dominance, but in continuity. They remind us that survival does not always favour visibility, speed, or growth. It favours fit. When we consider the possibility that entire ecosystems have persisted unseen for millions of years, the usual human urge to catalogue and classify feels less urgent, less central. Understanding becomes less about ownership and more about relationship, less about control and more about restraint. There is a humility in recognizing that some forms of life have thrived without ever needing to be noticed. Antarctica also invites a reconsideration of perspective. For much of this journey, the focus has been on what lies beneath the ice, what remains hidden, what we have not yet seen. But there is another layer to this story, one that turns outward rather than downward. From the standpoint of these hidden ecosystems, the surface world is the unstable one. Seasons swing wildly, temperatures fluctuate, light appears and disappears, species arrive and vanish. The surface is noisy, unpredictable and transient. Beneath the ice, by contrast, conditions are consistent. Time stretches, change is gradual. In that inversion, the hidden world becomes the stable centre, and the surface becomes the disturbance. Seen that way, our fascination with Antarctica's unseen life says as much about us as it does about the continent. We are drawn to places that resist us, that challenge our assumptions, that reveal how narrow our expectations have been. Antarctica does this not by confronting us, but by remaining largely unchanged in our presence. It shows us that life does not need attention to be meaningful, that existence does not require recognition to be successful, that entire evolutionary histories can unfold quietly, without spectators. As the planet warms and the ice begins to thin in places, this quiet may not last forever. Environments that were sealed may open, systems that evolved in darkness may face light for the first time. New interactions will occur, not by choice, but by consequence. What happens then remains uncertain. Some organisms may adapt, others may not. Some systems may transform, others may fade. These outcomes will not be sudden or spectacular. They will unfold slowly as Antarctic change always does, which brings us back to the present moment. Right now, wherever you are listening from, the world beneath Antarctica's ice continues on its own terms. Chemical reactions proceed, organisms feed, grow, and persist. Time passes without urgency. Whether we are watching or not makes a little difference to their immediate existence, but it makes a difference to ours. Because in learning about these hidden lives, we are reminded that our understanding of the living world is incomplete, and perhaps always will be. That mystery is not a failure of knowledge, but a feature of reality, that some things are not meant to be fully illuminated, only approached carefully. So if there is something watching us back, it is not a creature in the dark, it is perspective. It is the quiet reminder that life does not revolve around observation, that endurance can be invisible, and that the most resilient systems often ask the least of our attention. And now, as this journey eases to a pause rather than an ending, it is worth returning to something simple. Where are you right now? What does the world around you sound like at this moment? Somewhere far south, beneath layers of ice older than human memory, life continues without sound, without light, without witnesses. If any part of this exploration brought calm, curiosity, or a sense of scale that made the day feel a little smaller, then it has done what it needed to do. No conclusions required, no answers finalized, just a quiet widening of perspective. Antarctica does not rush, neither should we, so wherever and whenever you are, let the thoughts slow, let the questions rest without needing resolution. The creatures beneath the ice will continue their patient existence, and the surface world for a moment can afford to be just as still.